Yo, good morning. Good morning, gamers and gardeners. Although it's like good afternoon at this point. Oh, not the ducks! I need to adjust my hands so that I can better hold the ducks. Give me one moment and we'll get the dive on the screen. They just started part 2. We are in the Atacama. I think that's how it's said. We are in a trench. We are 2,837 meters down. And it's pretty cool. Literally, the temperature is 1.8 degrees Celsius. Which is almost freezing. Spencer, heyo! Disco, chocolatito, disco, chocolatito, disco, chocolatito, disco.
No sé si lo hemos hecho en español o en inglés.
Oh.
No recuerdo en ese tiempo.
Yo creo que ha sido en inglés. No sé si para nuestros amigos que estén en línea hispanohablantes quieres.
So this is another dive off the coast of Chile. So we are. It is a duo language stream. Hello.
Las dificultades que tienen en trabajar.
Sí, claro.
Apologies. Let me get this on the screen. Guys, I have maximum EPs. I am God's EPS soldier. There we go. Oh, the audio is still happening. Did the video freeze?
¿Cómo puedo llegar de ser un científico? Nunca sabía. Y más que cómo representa a los científicos en ciencia, es más como hombres. Hombres son científicos, científicas.
Let me refresh.
Para mí no existiera en ese tiempo Marte de Marie Curie.
Get it together, YouTube. Fish!
Estaba terminando mi doctorado y en mi doctorado estaba trabajando con una especie de bacteria muy interesante, Bacillus anthracis. No sé, probablemente muchas personas saben sobre esta bacteria que en 2000 en Estados Unidos tuvo un ataque de bioterrorismo de Bacillus anthracis. Se envió cartas con...
El anthrax, ¿no?
Sí, de anthrax, con las cartas con las curas de anthrax. Y eso fue un tiempo que el Bacillus anthracis vino mucho en luz. En realidad Bacillus anthracis ya existirá, pero mucho más en infección en animales tipo vacas, cerdos, este tipo de animales. Pero después de 2000 ya empezó esa idea de bioterrorismo. Y bueno, nosotros encontramos este Bacillus anthracis desde una región muy novedosa, desde unas aguas termales, y que encontramos que este Bacillus anthracis no fue un Bacillus anthracis patogénico. En los libros de microbiología nos veamos que está escrito que Bacillus anthracis que es un patógeno obligatoriamente. Nuestro trabajo fue el primer trabajo reportado con la biología molecular, con genómica de Bacillus anthracis, que no fue un Bacillus anthracis patogénico. Y también podemos entender que probable que este Bacillus anthracis para vivir en un ambiente muy extremo nunca tuvo un evento de transferencia de material genómica horizontal. Y también podemos confirmar con este trabajo que las bacterias patogénicas no vienen primero se vienen las bacterias no patogénicas. Con las evoluciones, con este tipo de cambio de material genética se cambia como una bacteria patogénica. Y durante este tiempo estaba buscando con los investigadores de Inglaterra quien primer reporte de Bacillus anthracis hizo. Yo postulé en algún financiamiento, pero no logré. Y en ese tiempo en realidad no logré ganar muchas posiciones de postdoctorado. Claro, en postdoctorado buscar posiciones es difícil porque en cada época es como un leaky pipeline. Gente se sale desde la escuela, después colegio, después pregrado, menos, magíster, un poco menos, en doctorado mucho menos y en postdoctorado mucho menos. Entonces cada fase hay algunas personas que salen. Y bueno, yo no logré ninguna posición en este tiempo en Europa. También tenía una dificultad que normalmente se hace la convocación de dar el certificado final del doctorado después de casi un año. Eso yo tenía un certificado como tipo profesional. Así que me logré, estaba buscando posiciones en Twitter, en redes sociales tipo LinkedIn, Twitter, y ahí encontré una científica y su paper anteriormente yo tenía leído y comuniqué con ella y yo sabía que ella estaba trabajando en Brasil, pero me comunicó que en este tiempo ella estaba en Chile trabajando como profesora y me mandó un concurso de una universidad en Chile, en región del Maule, Universidad Católica del Maule. Y ahí empecé a trabajar en 2018 como investigadora postdoctoral. Y en ese tiempo, bueno, en 2000, yo terminé mi doctorado el 29 de junio y llegué en Chile el 7 de agosto, así que casi no tuve tiempo de prepararme. Y yo pensé, bueno, América del Sur, vamos nomás. Y ahí tú vas a lograr todo en inglés, sería fácil, porque yo pensé, bueno, es América, pero América Latina es distinto. Llegando entendí que es muy distinto el mundo. Y claro, en el tiempo
I'm gonna turn this down a little bit because I don't think Twitch is
en 2019 entramos en pandemia.
primarily I don't think my Twitch audience I was about to say I don't think my Twitch audience primarily speaks Spanish, but there is the rocket jumper. Hello. We are once again off the coast of Chile in collaboration with a university there. You can see, I believe this is illustrating transit points. You can see the ROV right there, right there, and we will be moving along that. I'm not sure what this indicates. There might be a point of interest that we are going to check out. They are looking for methane seeps.
Después me cambié a otra universidad, la Universidad Autónoma de Chile. Yo pertenezco ahora de Universidad Autónoma.
I'm going to turn this back up and I'm going to step away for just a second. Yoni, hi, welcome! I'm going to step away to make sure the quail children are okay. Not the ones on the camera, the full-grown ones. Because they're chirping. They're chirping, and it could mean nothing or it could mean a squirrel spooking them. So I'll be right back. Let me turn this up.
La búsqueda fue muy difícil y el número de rechazos fue tan alto que el número de suceso o número de éxito fue casi no visible. Y bueno, ahora estoy trabajando como coordinadora de investigación de Universidad Autónoma de Chile, sede Talca. Y en realidad Chile fue un país excelente. Yo tuve mi vida como una científica en Chile. En realidad yo me estudié en India, pero mi formación completa como mi carrera científica de trabajo fue en Chile. Y muchos financiamientos tuve en 6 años, en últimos 6 años, 4 financiamientos. Y tuve un proyecto gigante de Antártica y 2 años fui en Antártica, lideré todos estos proyectos, lideré grupo y gracias a Dios fue todo bien. En realidad en India siempre digamos
Yeah, I can catch similar to Orange, one in every 30 words. It probably doesn't help that at least some of those words are probably technical terms. There we go. Sorry, and then the video buffered while I was away.
Gracias por compartir con nosotros tu historia en español, que seguro que algunos de nuestros oyentes...
Okay, wait. Okay, before I go any further, so we are watching Schmidt Ocean. We are seeing the live feed from the ROV sub-bastion off of the research vessel, the Falcor 2. And tomorrow, they will be doing a collab with NASA. Two of the people in the expedition right now are astrobiologists. So they're looking ahead at possible what we what we hope about here. Exploration on Europa or Enceladus. So that's very exciting. Tomorrow, check it out. NASA Q&A with astrobiologists. It'll be really cool. Okay, anyway, back to the dive. We're gonna send the ocean to space. Take that, ocean.
Getting in all the places.
When you think about it, you compare with the surface. It's like here you don't have
Schmidt Ocean and NASA is the Vtuber is my dream Vtuber collab.
Life is super rich. You can.
I said that as a joke, they're Vtubers, but also I would love to collab with them. We're gonna send Sebastian to space!
And almost the foraminiferas are everywhere, so we get a huge
Oh my gosh, the babies are squeaking.
It's like the grass, right?
It's like the grass.
Okay, sorry. Let's focus on the scientists. Here we go.
Yes, actually, this area, this coastal area of Chile is known for its high productivity, and that is what we are seeing. Almost looking into the sediments that we are working every day, the upper surface is actually not a soil. It's all the organic precipitation and loose organic material that we are looking into, and that is the reason when Sebastian is stopping and trying to collect a sample, we are seeing all those upwelling of the loose sediments.
So she's talking about the nutrient availability
also some
at
flocks
this
that we are looking
depth,
that are coming
because we are
through the
really
way of
deep.
Sebastian. Something is floating around there. There's something called marine snow, and this marine snow are formed due to some aggregates of organic material, some carbohydrates, some
Talking about marine snow?
decomposing material. So these are very common near
let
the
me turn
sediment of deep ocean.
There are a ton of sea cucumbers too. We're just flying over them, and I've missed the last like 3 dives. I think this is the 4th one. But this is a really cool area. They've been seeing Dumbo squid. They've been seeing lots and lots of sea cucumbers. It's poop. It's just poop. It's always been poop. The interesting thing about this is we're in the Atacama Trench, which is has offlow. There's a word for it, and my brain is stupid. But things flow from the desert into this trench. She was saying this area is kind of known for its productivity. So I mean, it's probably like mostly poop, but it might not all be poop.
They're like we are
this?
changing our turn now, and after me there will be my other colleagues who will be taking the charge. So we will be signing off now. In me, Apurna in dive stream, and Dr. Antonio Molina in sea logging. So we will be seeing you in some other opportunities, maybe tomorrow or day after tomorrow. And have a good day, everyone.
Yeah, Orange, they've been seeing
part
Jumbo
of the
squid.
world that you are looking into, and we had a nice time chatting about ocean life and the science that we are doing and our scientific trajectories. Así que chao todos los entes quien estaba observando el live stream y escuchando a nosotros. Yo soy Apurna, estoy hablando desde live stream y con mi colega Dr. Antonio Molina en Sea Logging. Estamos hablando sobre la vida bajo mar y también sobre nuestras trayectorias científicas y también en la área de ciencia que nos trabajamos. Así que hablamos en otra oportunidad, mañana o pasado mañana. Y gracias a todos para escucharnos. Que tengan un muy buen día. Chao a todos.
Thank you, Aparna. See you later.
Thank you. The video froze again, but the audio didn't. I might hit refresh again. Yeah, I'm gonna hit refresh because I don't want introductions to be happening while. Come on, come on. So this area has more poop than most poop areas, but also some not poop. Yeah. Man, I'll have to go and like find the clip that they sampled a huge sea cucumber, like big boy. If we have some time at the end of this stream, I think I wanna go see if I can't find them sampling it. can hear it, but it sounds like background noises. Yeah. Yeah, I don't think anyone's talking in the mic right now. I think they are doing.
All right. Hello, everybody.
Shift
My name is Yomar. I worked at Ambari previously, and I've mostly been on the background on this cruise, supporting science. Oh, there's a skate.
There's a skate. There it is.
I've been here mainly to support Dr. Lauren Seiler in cruise planning, especially with ROV operations.
Seiler is one of the astrobiologists.
I've done all the sampling sheets and helped everybody coordinate the projects. We have, I think, 16 different projects on this cruise. So guaranteeing that everybody gets their samples was a big part of it.
Hey, did you know in Mech Warrior 5 you can have a king crab?
We have Adam, who's piloting. Oh, they just had a shift change. Ben is co-piloting. And Carlos is our multimedia person.
Hello,
Then
Carlos,
there's
Ben,
Antonio,
and-
who's our geologist in the room. And Drew, who's sea logging.
Hello, everyone.
Carlos, could you switch Drew's mic on, please?
They cut this man's mic. The talking privilege is revoked. Can you turn his mic on? Everything goes quiet. No, we
had a
cannot turn his mic
question in
on.
the chat. Is
Yeah.
that a ghost squid? I think I missed it. I'm sorry, I just got
Oh,
here.
no.
All right, I think.
Am I? Yep, there I am.
Hey, Drew.
Hello, how are
Hello,
you?
Drew.
Hey, doing well. Did you get some sleep?
I did get plenty of sleep.
Can I hear you? Hi. No, let me
Yeah,
live
Drew
my
was on
parasocial
the early morning shift with me
fantasy.
here. This is an incredibly long dive. We're doing a transect. We have a bottom time of about 21 hours.
Oh my
The
god.
dive we started descending shortly before 5 a.m. and hit bottom at about 7, and we'll be staying down until 4 a.m.
Yep.
So we're trying to cover a large area to look for methane seeps today.
And then from 6 to 8, after we wrap up here, we're going to have a special graduate student takeover where a couple of the graduate students who have been with us on this expedition are going to take over sea logging and sort of dive leading the trip.
I think that's going to be a party.
Yeah.
Well, I hope some folks will stay up with us.
Yeah, we're going to be up till 4 a.m. I think we'll be taking turns.
Chat, I don't think we're gonna be
Lauren,
up that
I'll be
late.
here. So, in the hope of finding methane seeps. So we've been, yesterday and the days before, we've been mapping at night while people were sleeping. And then the day after, we'll look at the maps, we'll look at the bathymetry, and look for signatures or indications that there could be seepages. We're in a fault zone. We're at a convergent margin. In a subduction zone where the
So, yeah,
oceanic
subduction
plate, the
means
Nazca plate, is
it's going
subducting
under.
underneath the South American plate, and it's pushing it up. And Antonio, please jump in there. I'm no geologist, so he can probably explain that much better.
You're doing
And
great.
That subduction that's happening, that creates sort of like an opening for the methane to come through.
Yeah, the thing that's going on is like we have different kind of plates on.
I really hope not. If I am,
on the
I'm
surface
supposed to
of
be
Earth.
very busy. Yeah,
and
Scotty,
we
get
have
rest when you
this-
can. Yeah, my goodness. Gosh darn it. They're sending a signal from the bottom of the ocean. YouTube is the part that can't get its act together.
I don't know
There we
the
go.
name in English, but in the trust, I
Bro,
think they are called,
parasocial attachment to face
hiding,
streamers. Well,
below the continental plate. So in here we are right. in that part
parasocial
where,
attachment to
uh,
VTubers. Highest
this
spoke.
kind of
Parasocial
plate
attachments
that
to the
is,
disembodied
is,
voices.
is going down
Narrating the
continental
RSV
plate
streams.
that is
Me,
older.
literally me.
We are now in the
Nivek,
part of the
hi.
continental plate, and because of all these tectonic stresses, is why the methane is forming, because part of the mantle is diverts to the different cracks and tectonic features that are caused because of these stresses.
Antonio, sorry, one sec. We have a fishing vessel that is in the way. We have to go up to the bridge really quickly. Antonio, would you mind coming with me? Because we need a Spanish speaker to.
Oh, sure. I will translate. Sorry, Drew.
Coordinate. We'll be right back. Okay. Sorry, guys.
Oh no! It's pirates. This is now a first-person shooter stream. How do we know they're not just joint hallucinations brought on by stress and lack of sleep? Good question. Good question.
I don't think it really matters if I'm back here or up there.
Yeah, but I first started streaming when the pandemic hit, so nobody could go to school, and there was brief talk of me doing like childhood education videos. So it's just a holdover from that in my programming. You just gotta talk to them.
So
Can
while Jess and
you say
Antonio,
astrobiologist?
serve the fishing ship, I'll take over on the dive lead for a little bit. So I'm Drew Palmer. I'm a professor at the Florida Institute of Technology. We're about 30 minutes south of Cape Canaveral, so we're the people who get the best rocket launches, view of rocket launches anyway. And I'm one of the astrobiologists and biochemists and just microbiologists on the team, so I wear a lot of hats in this group.
Look at that sea cucumber. Look at that boy.
My group.
Why does the sea cucumber have a conning tower?
Hubrick and I are
What are you
here,
doing, buddy?
In part to look for methane seeps, but also to do some sort of comparative microbiology between the Atacama Trench, where we are now, and the Atacama Desert, where we were a few days ago. And that is one of the highest and driest and most Mars-like deserts on the planet, and we are hoping that we can use that comparison to help us understand some of the extreme conditions in which life persists, as a way of helping us look for life on other planets, like Mars. I just like saying Mars.
I'm glad that we have a geologist with the expedition to explain the tectonics. Unironically, there have been dives where they didn't have a geologist on watch lead, and there would be these really fascinating formations, and people would just be like, "We're pretty sure it's this, but we're not geologists." And I'm like, "I want to know."
I don't know, but they can communicate.
And we're back to our original positions.
Sea cucumber getting flashbanged by sea car. I think, yeah.
Thanks for standing by.
Why is a cucumber? You're a cutie.
We were
Imitation
waiting.
orange.
So we needed a Spanish speaker because I think the fishing vessel is somewhere along the track where we want it to go. So we'll have to communicate
Guys,
with
does that
them
look like a spiral
what
to you?
their intentions are. We might have to go around them. We don't really know yet. What is that?
Yeah, what is that?
Could we log
Mystery
that?
deep sea spiral.
What do I log that as? Oh,
Was it wire?
Adam said that's a wire rope.
okay.
Okay.
Anthropogenic.
Anthropogenic. Do sea cucumbers even have eyes or equivalent? I don't think they
seen that
do.
a couple of times during the day, and that makes sense.
Massive nautiloid shell? I wish.
I guess how it descends.
It sounds like it's a piece of wire that fell down. And the coloration we were probably
Yeah,
seeing
we're
is
about to
rust.
update our waypoint.
It
because
did look
we're actually moving
alive,
along
though.
much faster than we thought we would, so we can cover a bigger area. And we see more crinoids. Actually, we have a request from the science team. If we find another crinoid field, they would like to take a couple samples. So, and since we have time, we can do that.
So another interesting part about this area is, if I'm understanding the expedition brief,
Schedules and locations and goals are listed, so you can find the detailed descriptions of each expedition on the Schmidt-Oshner
Yeah, that's what he's
Institute
talking about.
website. The stream schedules are when the ROV is at bottom, which I think pretty much varies from expedition to expedition and is kind of subject to change. So, for instance, we made this decision to do this 24-hour dive, sort of on the fly. Not on the fly, but with a day or two of planning. So it wasn't what we'd originally planned to do. But
Right.
Based on the previous day's dives, it was a decision.
So the way this worked is we looked at the map yesterday that was acquired by the ship the night before. Oops. Ouch, that was the chair.
Yeah, I haven't before.
So yesterday we looked at the map and we have a couple of waypoints that we would like to go to, and depending on the structure of the sea bottom, we identified some geological features that we wanted to check out, and then in discussions with the tech team, which is amazing by the way, they assembled all the maps for us, cleaned them up, and helped us walk through a couple of different plans, and then together we came up with the dive plan for tonight that helped us cover the largest region possible. And so the logistics behind it always depend on what the other team needs. Will there be a CTD cast in the area? When does the ROV have to come up? When do we have to be in another spot? It depends on a really large amount of factors.
Yeah, lots
we
of
also
moving
have a
parts.
permit that we have to comply with. So there are certain areas where we can't go, so we have to plan around it, and come up with an ideal path for both Sebastian and the ship. But the crew is always helping us there, so and they've been incredibly supportive.
I think one of the more interesting things about this is that really the trip is sort of real-time data driven, right?
Yeah.
So we're
Yeah.
constantly getting data from
Like the
mapping
data, it's
and
real time.
making
Like the data they
decisions in
get
real time
influences
and
the dives they
adjusting
do.
our models and our hypothesis of
Yeah.
best places to look, which is really incredible to have the technology to be able to do that in real time.
Yeah. So we had a rough plan before that. We had a dive plan, but then coming here and the data. So there was a question in the chat as well. What kind of sensors or instruments do you use to help narrow down an area that may potentially have a methane vent?
Oh, we're going down.
We use all of the ship's instruments that are available. We used sub-bottom profiling to look at the sediment depositions, and sometimes if it's during shallower dives, if you get a really good resolution, you can see a certain fuzziness in the data, in the profile, and that can hint towards seeps, or you can see large sediment depositions. It tells you where rock formations are, so what the bottom looks like. So that is one part of it. Then we also look at what is called the backscatter, that is the acoustic returns, so we can see changes in reflection. And seeps, for example, if they are venting, which we don't know that these would be, they would have a very high reflectance because it reflects off of the bubbles. The other thing that we extensively looked at is the bathymetry, the map. So we used the ship's multi-beam to acquire data after the ROV dives.
Hello.
So
Sorry,
in
on
between
YouTube.
dive sites.
Jian, hello.
The techs help us to come up with a mapping schedule or with a mapping area, and that's ideally, and this all this data together informs where we dive, and how we plan the next dives. So it's very data driven because this is exploratory. We don't know where methane seeps are. We know that they were found north and south from here. And because it's all the same continental margin, there's a chance we'll find them here as well. And so we talked to a lot of geologists, we talked to collaborators that have looked at methane seeps in Chile, and they have helped us come up with the dive plan. Yeah, so we're looking for So it would show us if there were any bubbles, but we're also really relying on the visual. So we are looking for microbial mats. Methane seeps are bursting.
So nautical with the babies, yes.
And I think there's another lizardfish.
Oh! the video! Put the babies in the sub. No! Dramatic YouTube buffering! I hate you, YouTube!
Species we've been seeing all along our dives along this portion of the trench. That's I think that's the predominant crinoid species here.
Yeah, and crinoids can also be considered living fossils. They're very ancient beings, and you can also see them in the fossil record. You can use different species to date them as well.
So turning this down just a little because I want to catch up with chat just super quick. Orange says, I wonder if my hundreds of hours in flight sims and SnowRunner would be any good on a CV to apply for being an ROV pilot. Probably. That would probably... they unironically employ gamers. I don't know if they put the gaming on the CV, but...
There's this green name. I forgot.
Right, so the area they're in has been, if I'm understanding the expedition brief, the area they're in has been geologically stable for millions of years. So they're kind of in this area looking to see if they can find any living fossils. And as she said, crinoids, those sort of flower-like plants. Well, they're not plants, they're alive. They swim, it's terrifying. But also really pretty. Look, the crinoids, the flower-like creatures coming off the seafloor, would count as living fossils. It's pretty cool.
We'll find out if they're actively fishing or...
Orange says 500 hours in SnowRunner, followed by no moving violations ever. Instantly hired. Senior ROV pilot.
This is another really sediment-rich area. So some of the spots have been a little more barren, but this is very, very sediment heavy on these rocks. I logged them.
Oh, thank you.
It's quiet now.
I didn't even hear.
It was.
Oh, so we have a question. No sample collection in this dive. We were just discussing it. There is a request from some people in the team to sample some of the crinoids, but the primary objective of this dive is to cover a large area, so we will ideally hold off with sample selection until we find methane seeps. So we have a lot of space to take all the push cores that we want there. Because a lot of people
Jet Fuel says they're
on
looking
this
for
cruise, on
methane
our team
emissions,
are interested in finding seeps eventually.
which could indicate large
hydrates
biomass decomposing. But in this
associated
case,
rocks.
it would
Yes,
be
that's
the
right, we're
result
looking for
of
methane seeps.
tectonic activity,
But
methane breaking through the Earth's
if
crust.
there's something that a group member is very interested in, then we'll take a couple.
Is
of
B doing good? B
samples.
is doing good.
But yeah, in general, we want to cover a large area.
Well, I think we've found no shortage of crinoids today. Actually, there's
Yeah.
quite a bit of them.
Yeah.
What would you call an earthquake underwater? Still an earthquake. I know this is a joke.
Very true.
But earthquakes underwater are very, very much a thing and very scary. Oh my gosh, the little squeakers. Oh, it's not. I'm asking for real. I'm pretty sure it's still just an earthquake because it's still the Earth's crust, doing stuff, releasing tension. I'm not the team geologist, but yeah, tsunami fuel. Yeah, what do you call an earthquake underwater? Future tsunami warning. Yes, B. The little whale B is doing good. Poor Lambda is like 2 days behind. Are they really called sea quakes?
And that's a really nice field of
Also, Seth, hello!
crinoids. They're very plant-like in a way.
Oh,
Whoa,
there's a crinoid field.
that's really cool.
Oh,
That's
yeah.
beautiful. Can you please log that?
Yeah, absolutely.
So they're not. No, the dramatic buffering! I'm gonna. YouTube, please!
I normally try to count. I'm just gonna go lots of crinoids.
I've heard the term sea quake, but yeah, maybe I'm full of it. Maybe it is sea
Describe
quake.
it as a field of crinoids.
Yeah.
Yeah, those flower-looking things are crinoids.
Where's SpongeBob? We're just talking to Adam, who said they've got the ship and the ROV and Sebastian dialed perfectly, so they're moving along at the same speed.
Ah,
Because we're thinking about taking samples here, but if we'd stop, we'd have to play around with that again. So we'll, for now, just keep going, and there'll probably more crinoid fields later.
so for context,
Because we
we
might have to
are
wait for the fishing vessel.
2,000.
That's what he said. So that might be a good time for sampling.
There's also almost 12 hours left in the dive.
That's
So-
very true, yeah.
We're 2,800 meters
It's
deep.
a long dive. It's 21-hour bottom time.
No.
So please stay on later tonight. It'll be a party in here.
Everyone's gonna get exhausted.
Drew's gonna bring glow sticks. He
Yes,
promised.
I did.
I'll count on those.
Everyone's gonna be up too late and get really loopy.
Oh, we'll probably find something that glows. There's a lot of bioluminescence
Okay, sorry.
in the deep sea, by the way, but we can't detect it with our cameras. We would need a special low light camera. But there's I think it's a
Sorry, tell
BBC
him about it,
documentary on life that glows.
Mm-hmm.
They built Ben, my husband, built the low light camera for that one. Oh, there's 3 crinoids on one rock.
Oh, yeah, there. Okay, so sorry. I don't know how high Sebastian is off of the ocean floor, but Sebastian is 2,812 meters deep. So how is that, like roughly 3 miles? A little under 3 miles?
So there's a question in the chat about what we learn about methane seeps when we find them. So actually, one of the things we're actually
Okay, so
interested
just
more
under
in,
2
rather than
miles.
learning about the methane seeps themselves is about
How many
the
leagues
biology
is that? All right, Wolfram Alpha, save me.
ocean where there's no light driving biological processes and productivity. You're always trying to understand how life survives down here, and these methane seeps are sort of oases where there's a lot of activity.
It's only half a nautical.
going
Really?
on, at multiple scales, microbiological, so the bacteria as well as the macroorganisms. And so usually you find lots of sort of hotspots of biodiversity here. There could be microbial mats. There should be a lot of
Deepness, pretty dang. Yeah.
Jess was telling me that.
Yeah, I'm getting a similar,
so just tons of clams.
A similar response. Half a league.
The crinoids and cucumbers and sea cucumbers we've seen in bigger diversity. And so this is a hotspot area for looking for the biodiversity of the ocean, but also for understanding how microorganisms might survive in these environments. And so some
Yeah, that's.
methane seeps can provide us
Okay, so we need fathoms.
um,
20,000 leagues
for
under the
helping us
sea
understand rules for
lied
how
to
life
me.
might survive in other
It's the Kessel Run all over again. Joke's on us. It's also about half. It's also about half. Wait, no, that's one. 1 meter is a half a fathom. Hold on.
What is the question?
It was about the methane seeps and what we learned, so I was kind of giving the
Oh,
the
okay.
lowdown on the seeps.
Sorry, I was chatting with Adam about the fishing boat. He saw that there's a lot of evidence for fishing in the area. Oh no, crap!
Oh, it's because they went around the world in a summer. Got it.
Thank you. Yeah, so the rope that we saw that might have been fishing related. there was a little bit of trash too.
Writer Mark
So
Twain,
we'll see
Samuel
what we
Clements
find.
got his name from riverboat depths
It's all
measurements.
facing the same direction. I assume
So
it's
we
the
are
direction of the current, so they're
1,538
filtering.
fathoms under the sea. Will, welcome. 20,000 leagues was how far they went. Got it. I'm learning things.
Yeah, and crinoids are a very ancient group of organisms. They're also considered living fossils, and they first appeared about 300 million years before the dinosaurs. So they've seen some things.
They've seen some things.
Wow, this feels
Orange says I am not learning things. My brain is smooth. Literally fathomable. Yeah, I'm not sure how to get YouTube to stop doing this. It might be my rig. Let me shut down a couple programs that I don't
I'm
need.
not what I was just thinking about. I'm not sure if they can move their head kind of like a sunflower.
like a sunflower. Yeah,
can-
I was just thinking about the analogy.
because that would make a lot of sense, and I know that some corals can do that, but they're I can look that up. It's a very interesting question. Oh, there's a question. What are we looking to learn about the methane seeps when we find them?
Oh, that was the one that I answered.
Oh, that's
Yeah.
oh, okay. Sorry.
There you go. I think Craig ordered to you daughter cute kitty had smoothed her brain out. Unfortunately, oh no.
I tried to give the overall what everyone was interested in.
So part of the
A lot of
reason
sea cucumbers.
oh,
Yeah, there's a
a lot
lot.
of different colored sea cucumbers.
Many
Did you see the
cukes.
fancy one that looks like a little a little
Yeah.
punk sea cucumber
Yeah.
with the fancy hair? I mean, it's not hair, but
Yeah.
yeah. Part of the reason why we're here is Amando's group found microbes in the Atacama Desert. Those are really ancient microbes related to land plants. They're one of the oldest relatives of land plants. Oh, there's a little shrimp.
Little shrimp.
And genetic evidence showed that they were related to some microbes that come from the ocean. So
Oh.
part of the project will be to compare those with what we find here,
Yeah.
which could give us hints towards origins of life or land plants. in the
Seafood
ocean.
cucumber, yeah.
And they've actually found in some regions of the Pacific, I think it's below the gyres in the sediments, they found microbes that have been dormant for millions of years, and they can revive them.
YouTube's just cutting this. The knowledge is too powerful. YouTube can't let it get out. Hello, YouTube. Yeah, do not revive the ancient microbes.
That's pretty amazing. So the gyres are like a lot of the ocean is pretty much like the Atacama Desert. There's not much life there, but there's always microbes. But if those microbes don't find enough food to survive, some of them can go in a dormant state and survive until they find their food source, and they can grow happily ever after.
All right, I'm gonna.
Oh wow.
Okay, there you go.
I don't see them swimming very
Yeah,
well.
do not revive the primordial microbes. Worried about the ones coming from the melting polar ice
Yeah,
caps.
sometimes they don't like to be filmed. And then they
Life
take off.
finds a way. to revive the primordial microbes that went dormant deep under the ocean.
Very nice. So one of the questions in chat about acorn worms, yes, we've seen quite a lot of acorn worms down here.
Should I try this in a new browser? Hold on. Just give him a cookie and some orange juice. He'll be right as rain. Do not! Do not! Also, Will, I have stories to tell you about these stinky little quail and the stress they have been giving me. Oh boy. Not right now, though. Right now, I'm fighting with YouTube. What's your problem? What's your problem? I would like to watch the live stream, please.
But I still don't know if the stalked ones can move their arms. They probably can, I'd assume, but
It does have different resolutions. I'd prefer not to nerf it though, because I'm honestly not sure that's what the problem is.
yeah, I'm more of a microbiology person. My marine biology classes have been a while ago.
Midnight, hello.
So you're a marine microbiologist. So.
Yes, yes. But in the past, I've also worked with cuttlefish and cephalopods. But I think at that time, I was considered more to be a neurobiologist, or
Everybody
that
sit
was
up
actually
straight.
during my undergraduate studies.
Oh my gosh, the
I
babies are
was
also
studying
waking up
biology
to
and then
calibrate.
later biological oceanography, with a focus on microbial ecology, and this is where I've done most of my work in since or after my undergraduate studies.
And if
And
you were
I-
sitting on your legs, do some duck steppies.
We went to methane seeps in the Monterey Bay.
And
Those are
relax.
false seeps that are produced by organic matter deposition.
Okay, so you can get methane seeps from
Those
organic
are
matter.
sometimes very
Horrifying.
short-lived. And so the one year we went there, that must have been 3 years ago. They were active. They were bubbling. We saw lots of live clams, and we did incubation experiments on, I think it was in 1,000 meters roundabout. That was right by the canyon. And a year later, we wanted to recover those cores, and the whole clam field was dead. The methane had stopped seeping from that area, and it's probably moved because we found other areas where there were still live clams. But it was incredible to see how fast such an environment can change, how rapidly.
Some, I think the term used was extinct
Yeah.
methane seeps, where
Part of decomposition from
that was
particularly large
intriguing because you
biomass
wanted to look at
deposits. Yeah,
what's
yeah.
essentially the microbial population that remains after that, and then some of the macroorganisms there as well. But today we're looking for the live seeps.
Yeah. And we're also looking for the live seeps, but having found evidence for ancient methane seeps already is super exciting, because that means we have been looking in the right area, but we're still waiting for the final confirmation from
What are you,
from the
jellyfish?
data. What?
Oh!
that?
Oh.
Was that a little jelly?
So they're not stopping because Sebastian is tethered to
So I think they will
Alcor
look
2,
at the
and they're moving at the same
the
speed.
isotopic carbon composition to see where the
But if one were to
so we
stop,
found
it would throw
what
off
is
that
likely
rhythm.
carbonates. They will look at the composition, and then they'll find out if those were actually methane seeps, or where they came from, whether it was from organic matter deposition.
Hold on. While they talk, I'm gonna see if the babies need more wet mash. I'm giving them wet mash to encourage growth because one of them just hatched and the other had a rough hatch. Hold on.
But today we're looking for active methane seeps. So we're mostly looking for visual indications of methane seeps. For example, microbial mats, and we can see microbial maps usually by discolorations of the seafloor. So sometimes they appear white, and we looked at pictures from methane seeps that are south from here, near Concepcion in Chile, and they're whitish, almost blue along corridors. We identified regions of interest, and this is why we picked this dive site today, and we're going to cover a really large area within
Not a phone in sight.
21-hour bottom
just
time.
people living in the moment.
Dive will be until 4 a.m. and hopefully lots of people will stay up with us.
Just a reminder, Jess and I are going to be on until 6 Eastern Standard Time, and then we're going to have a couple of the graduate students who have been working so diligently on the samples we do bring up. They're going to take over and do the data logging and dive lead for a couple of hours.
It's worth noting,
Dumbo?
we're moving quickly, but they have access to...
There was a Dumbo octopus, I think.
Oh! It's the small red thing right there.
Hey. Oh, it's a tiny one.
Yeah. Right there. Ah! Look at the little guy! Look at that little lad!
Get that in focus. Aww.
YouTube, no! Alright, I'll drop the resolution a little. They are quite deep. And in the middle of the ocean. Look at that lad! Let me turn off the quail cam since they're sleeping anyway. There we go! There we go!
Aww.
Look at that boy!
A little Dumbo.
Yeah! The big ears. Oh, that's a wonderful critter. Let's give him a little
It's very
poke.
cute.
Oh, that is an adorable octopus. Let's go.
Yeah, this one is much smaller than the one we found earlier. Ben just reminded me that was the one we found earlier was, I don't know, maybe 5 times the size of this one.
Oh gosh. Yeah, that was a good eye on them,
It's
catching
really hard to
that
tell
as they
because
flew over.
distances and sizes.
Yeah, scale
Are-
can be challenging.
Yeah,
Octopi definitely
scary.
fascinating. Such
Yeah.
interesting creatures. Yeah, they're very cool.
Yeah, going back to the students, huge shout out to them. They've done
I'm gonna try to turn it up
amazing
again.
with all the sample processing. They've been collecting all the water samples, filtering all day long, taking the core samples, organizing themselves. It's been amazing. Thank you guys so much.
Worst case scenario, I'll back us up like 30 seconds. Normally the Takodachi watches the VTuber. Yeah, not the other way. Ah. What a time to be alive, where Teeny Octopus can be streamed across the world from a ship smack in the middle of nowhere. Isn't it great? Smack in the middle of nowhere and nearly 3,000 meters under the ocean.
Remind me,
Almost
Drew,
2 miles
what are
under the
you
ocean.
interested in the methane
Crazy.
seeps? What are you looking
Sure.
for?
So we are predominantly more interested in the astrobiology component. So these are Methane seeps are the interesting spots of biodiversity. Methane-related microbes were probably prevalent at one point on the surface of Mars in an earlier time point. Part of the geological history of that planet involves methanogenic microbes, and so that's one of the things we're looking for is signatures for those organisms that live in these kind of environments that then we could extrapolate for helping us detect the presence or former presence, so the after effects, the shadows of microbes that lived on Mars in the past. But we're also just generally interested in these hotspots of biological diversity in rather challenging environments, because that can create really interesting metabolism, chemical processes that we can mine for a variety of potential benefits, right? So there may be new antimicrobials down here, new antifungal compounds, different types of treatments that could be mined out of this environment.
That's really interesting. Yeah, fingers crossed that we find them tonight.
Yeah.
And if not tonight, maybe tomorrow, because tomorrow we have a similar dive with less bottom time though, in a slightly different spot. I think it's 20 kilometers from here. It's closer to shore and a little bit shallower. We're kind of moving slowly over there, but we won't reach this point on this dive. But there's another chance of finding methane seeps if we cannot find them today. But we might have found ancient methane seeps that used to be methane seeps at some point, but are not currently active anymore. But our geologists can hopefully tell us more about that once they analyze the data.
There's a question in the chat about Martian microbes and methane. There's some really good work. There's
I keep wanting to pause to address chat, but this is all good stuff.
Sugar, SCHUG. Nope. Andy Sugar. I don't have his spelling, but I'm gonna get it wrong. I get it wrong every time, and I'm not sure. Some people call him Andy Sugar, and some people call him Andy Sugar. So I know it's not SUGAR, but Andy's done a lot of really good work looking at Earth extremophiles surviving in Martian environments. So that's some of his work. So, and he's done a series of papers looking at methane microbes from Earth and using those as proxies to try and understand maybe the past geological history of Mars.
Okay, so I'm going to turn this down for just a minute so I can catch up with chat a little while they fight with their own displays. So Rival asked, how do you get a 3D scan of the ocean floor? Because a moment ago there was a scan of the floor that was showing the model of where Sebastian was. And the answer is they took that scan the night before. They took that scan and they were looking for evidence of methane, and they did find some. So we're just kind of making our way downtown across the area that was scanned and looking for visual evidence of methane seeps right now. The Okeanos Explorer, the Falcor 2, and the Nautilus are all part of an initiative to get really detailed maps of the seafloor. These live dives are just kind of like a cherry on top of that. But yeah, that's how they got it. They made it, and then they're like, all right, let's go drag the boat around. Let's go drag Sebastian around on that. I hate to be that cat, but Sebastian's not the one broadcasting. True, Sebastian is sending all of this back up to the ship, though. I think literally in 4K. Cable may affect time. This is something I heard about. I believe it was the Nautilus talking about this, so other ships might have other setups, and they did eventually fix this, but there is absolutely latency between the video and what they're seeing on the ship, between real time and what they're seeing on the ship. Apparently there is not latency between the operation of the claw and what they're doing on the ship. So you're operating the claw in the exact time, but then you don't, like, there's a delay on the video, which is crazy. It truly is gamer suffering. What is up, fellow underwater enjoyers? Yeah. Dumbo caught in 4K. Yes, we caught the Dumbo elephant or Dumbo squid in 4K. Sebastian feeling cold? Does he need a blankie and some hot chalky? Sebastian is in water that is 1.79 degrees Celsius. So Sebastian might want a blankie and some hot cocoa when Sebastian comes back up to the ship. Data takes roughly 4.6 nanoseconds per meter over copper. So Orange wasn't kidding. You could literally do the math. Okay, I will turn this back up. They're talking about a lot of cool stuff. There are astrobiologists on board. They're like relating this desert and this subsequent area under the ocean adjacent to the desert with like life on Mars. We're getting a close up of a lad.
Look at its eyes. Oh, it looks like a little alien.
Oh my god,
It's an alien octopus. Oi.
Oh god.
Cute.
Similar data electron transfer is very similar thing to speed of push. Look at that boy! Look at that
And is
boy!
that a brachiopods
my gosh.
right next to it?
It's one of those snails. Hi, friend.
Oh.
Oh, there
Oh,
it
wow.
Bye-bye.
goes! Oh,
Can we zoom in on? Okay.
I think we're gonna follow the friend.
Stay long enough for me to get it on camera.
Deep dweller encounters light. Yeah. This time we're the ones being the alien encounter. There it goes. Nyum. Oh, bye, little one. Deep sea a lama. Oh,
we
yes,
have a
yes.
question. After taking ocean floor samples, do we have to maintain pressure levels in the lab? No, we don't. What we do is we freeze the samples. Ideally, what we did in the past is and it depends who you ask. So for microbiology, we usually, in the past, we snap froze them. Unfortunately, we didn't. This time, we didn't have liquid nitrogen on board. So what we used was some chemical compounds to preserve the DNA that is in there.
Different.
And then we just mix it and freeze it. And then it stays intact for quite a long time. And then we can take it back in the lab, extract it with some different chemical processes. And there are lots of steps after that. So taking samples is one thing, but then it is month of work to actually
processing samples.
extract everything, send it out. Then there'll be an organization that usually does the sequencing, unless you have your own machines for that in the lab, which a lot of people don't. So we send it out, and then we get the data back, and then the fun part, the analysis starts.
Yeah, from our water column and our core data, we'll be doing microbiological and chemical analysis. And the chemical analysis could take a really, really long time.
Yeah. How long does your analysis usually take?
Well, so we've never analyzed samples like this before,
Usually
so
say
that's going to
curlies.
be,
And
in a way,
that
we're
didn't
going to be
quite look
doing
like
a very,
a curly.
taking little bits to start with and kind of getting down the samples. So we've done environmental sampling before, so we're not, completely foreign. The water sample analysis will actually not be that hard. What we're doing is, rather than bringing too many water samples home, we can take about a liter of seawater that we collect and filter it through these little cartridges we have, and those will trap most of the kind of biomarker molecules that we're looking for, what we call the organics. Then we can take the cartridge home with us, and the cartridge is like, I don't know. I'm of course realizing that holding up my fingers is not a good way to. So it's like 4 inches, 5 inches, and And those
That's like 15 centimeters. Oh, I think there's some trash on the left-hand side. Did you want to lock that?
yeah. And so then we can
There's a
take that
Discord
back with
for,
us. So instead of taking like
dive
50
streams.
liters of seawater, we're gonna have like 30 cartridges, and then we
which
can
let
just
me see if
drain
I'm
those
able to
off
generate
with
an
some
invite. I'm not sure if I
alkalinization we have.
can.
Okay. And is that all going to be Katie's
I'm in it,
job?
but
Do
I
you
don't
have
like-
more
I'm
students
just
in
in it.
your lab?
So right now, I think Katie's going to be sort of spearheading the
Yeah.
discovery process, and then we'll see like how much we have to work with, right? And then she might get a spin out
It
a
froze because
cohort
Discord
of
opened. It's not frozen.
students on the
There we
project.
go.
This is really why, for us, finding a methane seep to compare all this to would also be really exciting, right?
Yeah.
So.
Will you
I
also
think-
be?
Oh, go
sorry.
ahead. Go ahead.
No.
All right. So I was going to say what we've already found, right, even in the
Yeah.
absence of an active seep, there's a lot of interesting things for us to
I
look
should
at.
be able to, but I'm garbage at
that-
Discord.
That I think is going to keep us, intellectually engaged, so to speak.
Yeah,
Um,
that's-
But yeah. So
Would you be comparing it to the ancient
Okay,
methane
this is
seeps
not
if we
my
have- if we
Discord.
get
This is the
confirmation?
dive stream Discord.
100%. Yeah, that's absolutely
That
kind
should
of
work.
what I was thinking about last night.
Yeah.
Was that's really what would be the goal would be to compare places where we cored at the very beginning where we really had no evidence of seeps.
What in the world
A
is
place
that?
like
Who are
yesterday
you?
where we thought we saw some ancient ones.
Oh, an enemy.
And then
Maybe.
following up with an active seep, I think that gives a great range. We also collected some interesting profiles along the water column. So through the oxygen minimal zone, which is sort of a high productivity region, and down here to
Makes
the,
sense as
the
a
floor.
muso
So we can make some comparisons
Moose-
about the
moose
microbial
octopus,
populations
as
and
we are
metabolism
dealing with the
that's
seep
going on
area. Ah.
in those environments, which again, that in a way actually contributes to how we think about biosignatures. How do you
And I know you're
characterize the footprint of biology in really, really rich environments? It's very easy to do. And then as you get to more and more sparse environments, that becomes a more challenging goal, a more challenging issue,
Did you, you guys went to the Atacama Desert, right?
Yes.
Before we went on this cruise, did you take any samples there, and will you be comparing those as well?
We did and we will.
Oh.
Yeah.
So we got a couple of interesting rock samples from a couple of different sites in the Atacama, and we even found a couple of
Squad leader, hello.
rare, hard to find water samples. There is water in the Atacama. It's just in very specific places. And then one of the sites that we went to that was
You guys
really
should have stream
fascinating
avatars, I'm
was
sorry.
pretty high up, and there it does not rain, but it looks like, we were talking to Armando, every morning it's like clockwork. There's a fog comes in, and then it lasts for a couple of hours, and then gone, and the sun beats down, and like the temperature goes up like 30 degrees Fahrenheit, 30 degrees Fahrenheit. It goes up. It gets, and then the fog's just gone. But they think that during that time, we found green on the ground, right? That was not
Yeah.
copper, and so it's a pretty good indicator that that moisture in the air is enough to sort of give these organisms a sufficient amount of water. Then they get the light, they can do some photosynthesis, then they
Yeah.
get into the shade, right? And so they have a very short period of time based on how some of them are on the slopes where they would get both moisture and sunlight, and then they'd be out of the radiation. So the UV in the Atacama was also really high, which is also problematic. And there's some models on from a sneak in Mars. There are models on Mars for different surfaces that might go through like these kind of freeze thaw periods where ice is, most of the time you're not going to have liquid water on the surface. You could get brief periods where sun shines on a particular spot on a crater that would allow that particular amount of water to melt. That could allow the biology to run for a little bit, and then the sun goes by and that stuff gets cold. You could have these freeze-thaw cycles, which has actually been proposed in Origins of Life chemistry. Sorry, I got going.
Oh, yeah, I know. I'm processing.
So basically, in this
So the
desert, there's no rain,
freeze-thaw
but there's
cycle
fog. And a similar
in
process
my quotes,
might.
I missed that part. Can you?
Yeah.
Whatever.
So the idea would be that one way that the microbes could persist, right, is that they
Yeah.
basically go into a
Desiccation?
in a frozen state, right? It may not be completely dried, although, liquid water on the surface, the pressure on Mars is so low that
Listen to these nerds nerding
at
out.
room
Isn't it
temperature
great?
pressure, liquid water would not last on the surface of Mars. It'd literally go into the gas phase, like just evaporate right off. But the temperature is much lower on the surface of Mars, so it's possible that there could be standing water very briefly under the right conditions, and so it could be that water briefly goes into a liquid form and allows those microbes to kind of do biology for a little while, and then they freeze over again, and then it thaws again, freezes and thaws, and they could kind of survive through these kind of cycles like that.
Interesting.
Yeah.
Would they also use glycol in their cell plasma, in their cell fluids?
Oh, so
For
like as
antifreeze?
Yeah, as an antifreeze. So that's possible. They're also finding nowadays, and this is actually something that Armando and I have been talking about for the bacteria that live in the Atacama, is there's a growing body of research that shows that a lot of microorganisms that live in these kind of unusual water conditions where water can suddenly be gone or unavailable, they maintain these things called coacervates, which are like almost like oil droplets inside of the cell that's made of amino acids or other materials, and that allows them to make almost like an antifreeze. And they're interesting because they become their structural properties are really interesting and kind of the reverse of what we normally see. They've been found as these kind of proteins have been found in tardigrades actually as possible protectants. And they think it's one of the things that allows tardigrades to survive.
I love tardigrades. Little gummy bears.
Yeah. Everybody loves tardigrades.
They're great. They are great.
Yeah, and those microorganisms that they found in the Atacama Desert, they also had, because we had a question regarding viruses, they found evidence for viruses in those, and those had a marine origin. So yes, we will be looking for viruses. There is a person on the science team who unfortunately couldn't make it, but we're taking
Could they not
samples
make it
for her.
because of the viruses?
And she will be looking for giant viruses in the water to compare those to the ones they found in the Atacama Desert. And it is true, we're not very far offshore. We are
I know
currently
logically
about
that this is not a horror
43,
movie or a horror
45
game. This is
kilometers.
100%
That
how the
is
horror game
nautical
starts.
miles. Around about 24. We're in a subduction zone looking for active methane seeps. I don't actually know the answer to this question. Is there a public catalog of the usual suspects of fauna we're coming across?
My assumption would be no, because this is an unexplored area, really. So
Yeah.
we're cataloging it for the first time. There's a few of the marine biologists on our group who are we've been taking samples the last couple of days, and then there's going to be 50, 60 hours worth of video to peruse for identification. So I think with that, maybe they'll be able to kind of establish a diversity of at least this portion of the trench.
Yeah. There might be a Schmidt catalog. I think maybe we can ask Hannah that question because she's in the room. Hannah, is there a catalog for common species from Schmidt?
Not that I'm aware of. I think it depends on where you are in the world. Because while these are common deep sea species,
D68H.
we don't
It is a
know
lot of work.
some other common deep sea species elsewhere.
It is a lot of work. Something that happens on these expeditions is they end up
So Hannah was just saying that
with a lot of data.
some of those species are very common deep sea species that we find in other areas of the world. So there will be a catalog for those. There's none specific, or at least from what we could find, there's none specific for this region yet. But that's part of the reason why we're here. This particular area of the coast of Chile hasn't been explored very much. So in the last couple of days, during the night, we've been mapping the area and then deciding based on the map where we wanna do our dives.
I would add that Esmoy, which does have some catalogs.
The where?
The Ocean Institute doesn't. The Center for Ecology and Sustainable Management of Oceanic Islands, or Esmoy here in Chile, does have catalogs. But they're a little more specific to the Salas y Gomez Ridge.
Oh, it's more specific to the ridge?
So I'm gonna turn this down just a little bit. Birdie says it takes me 3 to 4 hours to go through 1 hour of footage. Yeah, it's a lot. It's a lot. Someone is going to have to come through and look at all of this. So when they do these dives and they get all of this video in even higher quality than what we're watching, someone has to go through and look at all of this information. And there's actually a really cool game out right now. So it does use AI, and a lot of the fuss about AI is that the data banks that it uses to learn are unethical, like they're scraping artists' artwork. But you can help build the data bank by playing this game. Yeah, Fathomverse. It's Fathomverse. It's by Mbari, or in conjunction with Mbari, the Monterey Bay Aquarium Research Institute. And it's just a really simple little phone game. It's on Apple and Android. And you just identify fish. You just go through and you do some trainings, and then you're like, yep, that's a fish. And by doing this, it builds up a data bank that will hopefully be used to train an AI to identify things faster. Because legitimately, there is a lot of data. There's so much data. But yeah, it's really cool. I work at a different thing, but also with AI. Really? So is this just kind of, I mean, you might not know, or you might not be able to answer. Is using AI kind of a
Well, I hope you'll
kind of a thing that's gonna
the late night show.
that's emergent in that industry as well?
Yeah, it's gonna be like,
With deep
it's
sea
gonna be
deep sea critter recognition
Glow sticks.
starting. Yeah,
Glow sticks.
that doesn't really surprise me. That's, I don't know. I think it's kind of good as long as you have a person double checking the work. They're outsourcing the work. It's true. It's kind of exciting though, because again, like with generative AI, the issue is the data sets it's trained on aren't necessarily ethical, but you can help build a data set. You can contribute to science. Ethically training AI. AI trying to recognize animals is how you get AI obsessed with round birds. I have no further comment. I have no further comment. But yeah, it's really neat.
Paperwork.
Yeah, these dives are so cool. Like this expedition specifically, all of these dives are cool. But there he goes. There goes Green. There's a lot going on with this one.
So Ben, co-pilot
They are very
right now.
adorable.
So sorry, they had a shift change. We didn't mention that. He just made me aware I have a funny accent. I almost sound Canadian. I'm German, but I've only once been to Canada, and I crossed the border, and I forgot my my little dog's paperwork. So you, in order to come back to the U.S., you need to show like the vaccine record, and I had remembered the day before, but the day we crossed the border, I forgot. So I crossed the border, and the officer there told me, or asked me, do you have your dog's records? And I said, no. He said, well, you can't go back. So kind of panicked a little bit. But they sent me, a very kind friend sent me a photo of his vaccine records, and then we walked, after a couple hours, we walked back over, so we went to the Niagara Falls. I spent a couple
Not
weeks
very good at spotting accent
in,
locations. Got it right, yeah.
Upper New York State. So one day we did this excursion, and then on the way back, the officer on the American side didn't even ask what it was, so I panicked.
No. Smuggling that dog across the border. It's a very faint accent. It's a very faint accent.
Come on in. Which is usually not the case, by the way. I've been interrogated many, many times at the border. I don't know what it is.
This sus German lady and her sus dog. Why does the seawater taste blood? But it's amazing when the same water ends up in the Himalayas, because it goes
Currently
through
live on a sailboat in Mexico, so we're traveling quite a lot for work.
There's the salt water.
So I think it's all the stamps in the passport.
Doesn't get evaporated. Yeah, the answer is salt. Although, isn't the trendy thing right now Himalayan salt lamps? Why is the Himalayan salt also better?
No sleeps till seeps. Yes, yes, I like that.
Oh, no
small
sleeps till
coffee.
seeps. Yes. Because pink, a solid proposition. Hawaiian sea salt also pinkish or reddish, also superior. Yeah, look at that anemone.
Did we have a chance
That's just a robust
take any
lad.
of those crinoids, or are we waiting until later?
We wanted to wait
Himalayan
until we
sea
hear
salt
back
really
from the
does have
fishing
a
boat if
different
we maybe
flavor.
have to
I'm not
stop
sure how to
or,
describe it.
Oh, okay.
I think it's because the rock salt is also purified.
or diverge, or if we have to spend some time
Yeah, I think
somewhere.
the minerals would be
Um,
different, right? That
I
would
know we
alter the
crossed this
flavor.
one beautiful crinoid field. We were hoping in the next 12 hours there's probably another one.
Yeah, we'll probably see more.
So when we figured, have you heard back from the fishing vessel? Can we still proceed as planned?
I did not know we were waiting to hear from the fishing vessel.
Oh,
I just stepped in, so.
They left.
Oh, okay.
They
Oh, okay.
left.
Oh, that's nice. That's very kind.
Roman, hello. Yeah, there was a fishing vessel. I guess they were like headed pretty much right towards it, and they weren't sure if they would have to detour or what was gonna happen.
So yeah, we will update our waypoints. We're making really good speed. We're much faster than we anticipated. So we will cover a larger area and check out some of those ridges and fault lines in the hope of finding methane seeps.
So
We're
when I
also
was
looking at the,
skipping ahead,
Sebastian sonar. So if there are any bubbles, which we don't know
what?
that there are, they would show up on the sonar as really high reflection, really high returns. But we're primarily going with visual indications. So we're looking for microbial mats, so discolorations on the seafloor, or specific animals such as clams. So sometimes you can see those clam beds, or tube worms. We might have found ancient methane seeps a couple days ago. I think it was 2 days ago. We were waiting for the geologists for their analysis to see if those were actually methane seeps. Some of those rocks looked like carbonates, but they haven't confirmed that yet.
Oh, enjoy your lurk. Thank you for popping in, Roman.
Oh, so we sailed. There's a question: where about in Mexico? We sailed in the Sea of Cortez. We left last year from California and sailed down the coast, and now we're spending some time in the Sea of Cortez, which is very beautiful but also very hot.
Mm-hmm.
We spent some time on Isla Isabel. That's a marine protected area where blue-footed boobies, brown boobies, and
jealous.
frigate birds are nesting.
I like how
And they don't even mind
high
you're
security
there.
containment cell also looks like
They
it's
don't care.
underwater.
Take really great pictures.
And the
Then
heat in the
if you
ship is
go
very
there,
different than anything you'll feel
the
on
birds get
land.
really upset
The
with drones.
humidity must
So
be
they're
crazy.
more upset about drones than about people visiting the island. Unfortunately, they don't allow dogs though. And there's a couple of fishermen living there, and they make sure that you don't bring your dog on the beach.
Don't laugh at the
So we
movies.
didn't stay for too
They're
long.
very silly though. Also, Pavlov,
There's a
hi.
question about how do you tell the difference between an ancient seep and an active one?
Great question. So an ancient seep, you can still find the carbonates and the carbonate depositions. Those were the shelf-type structures those might have been the shelf structures we saw.
I've been on a couple warships,
but
and the
no-
combinations of metal at sea humidity can be
None
oppressive.
of the species,
Oh, I
none of the
bet.
microbial mats, nothing that hints on this oasis of life that methane seeps usually are. So methane is used as a food source for lots of different species, and those would be the ones we would expect to see there, like microbial mats, riftworms.
I imagine even in a ship, like they're not going to be designed to.
Permanent.
Yeah.
They're actually pretty transient, so
Yeah.
they can run out of methane, and then the species run out of energy and food, and so
Yeah.
they just kind of leave a fossil behind.
Yeah, I was saying earlier we went
See,
to
this
the
whole
methane
time I
seep
thought
in
the methane
that must
seeps-
be 2020. And it was active. There was a clam field. We took samples and we left them there for incubation. Initially, we thought they're going to be there for 6 months, I think, and so we had another cruise planned, but that got delayed because of COVID, and we went back 18 months later in 2021 sometime. Recovered those cores, and by the time we recovered them, the methane seep was dead, was not seeping anymore. All the clams had died off.
Oh gosh.
So we think it probably moved somewhere else.
So my question is, when we say they're transient, are they typically transient on
It
like more
ceased to seep.
geological timescale, or like more of a relevant biological timescale? I mean, relevant for like what I do.
Both. So it depends what kind of methane seep you're looking at. So the ones that we were sampling in Monterey Bay, those were some call them fall seeps. They're there because of organic matter deposition. So it's a lot of organic material that is being piled up. And
Leo, I'm going to
there's
address that
degradation,
actually. 1 second.
And
I do want to
decomposition
hear this.
produces methane that other species will use. And the ones that we're looking for here, well, we'd be happy with either one, but there's tectonic ones, and a lot of those will change on geological timescales.
Okay.
But
So
it
there are
always depends.
tectonic
So
ones.
different regions have, it
So then,
depends.
is it? Sorry, I think
Yeah,
it's
no.
not intrigued. So is it possible then that a previously active methane seep can produce sufficient productivity that after it runs dry, essentially could create a fake seep by virtue of the organic matter?
Oh, wouldn't it be diminishing
The
returns
real
though?
seep leads to a fake seep.
Interesting question.
Yeah, that's a
the
good
answer is I don't
question.
know.
Yeah.
Maybe, I wonder if maybe Antonio knows.
Maybe Antonio knows. Yeah.
So
I feel like that's one of those things we can't know until we see it happen.
A real seep that's driving biological activity. When the real
I'm gonna turn this down while they debate that. Okay, so Leo says billionaires are idiots. They could buy a submarine and explore the sea forever, and they choose to buy Twitter and be idiots. Okay, guys, come here. So there's
Maybe.
there's a oh gosh, what is it called?
Yeah.
There's. Give me a minute.
Not an
Just give me a
expert
minute.
on methane seeps. I just did a little bit of microbial studies on them in the past. But this methane seep hunting for me is.
What was the name? What was the name
We learned
of?
a lot in the last couple of months in preparation for this cruise. We were looking at data from seeps north from here and seeps south from here, and tried to compare that. This is how we picked the dive sites.
I mean, obviously, this might also be a complicated question to answer
Yeah.
because this. With
The limiting factor. Okay, all right. So Leo says.
everywhere in the
Leo
ocean,
says.
it's being hard to find them. You have to find
One moment. We're just going to ignore James Cam. We're not going to ignore James Cameron.
Yeah.
Okay, so gamers, gather around. So, Leo asks, billionaires are idiots, and they could buy a submarine and explore the sea forever, and choose to buy Twitter and be idiots. So, there is a submersible, I think maybe one of the few, possibly the only submersibles that can currently reach the Titanic. It's called the Limiting Factor. Its creation was just this big, dramatic, cooperation of several organizations, and it's this really impressive piece of work. Do you want to know who owns the Limiting Factor right now? Gaben. The Steam guy. Gaben owns the Limiting Factor. He also bought a company called Inkfish Ocean Exploration. I don't know if they're gonna do live dives. Their website says coming soon and has said coming soon for ages. Gamers, we're going to the bottom of the ocean. Get your Xbox controllers ready. We're going into the ocean. But yeah, it's really cool. It says no. Steam Link. Yeah. Sorry, the thing paused because I got big. Let's go back into the stream. He is pretty based. Didn't he use a game controller for it? So here's the thing. If we're talking about the sub that imploded, they were using a Logitech controller. However, that was like the one thing that wasn't sus about that whole arrangement. It's actually pretty common to use game controllers to pilot ROVs. What are y'all squeaking for? The Titan, yeah, that was like the least sus part about that thing. Steam Ocean Expedition Edition. Yeah. Look.
It's on the different layers, and they did
It's
an
just really cool.
amazing job.
Can't believe gaming is training us. Yeah. Real life based Subnautica win.
What age classifies
man.
methane seeps as ancient? I'm not a geologist. I would pass this question on to.
He left the room.
Oh, he left the room. I'm sorry. We don't have a
The
geologist.
geologist left.
If we can remember,
Under
we
the
will ask
sea.
him when he comes back.
Under the sea.
I think there will
Some
be a
billionaire
geologist
from
on
Ohio
there.
wants to join.
You're
No,
coming.
no.
Yeah. He escaped. So there are differences between ancient seeps and newer seeps or active seeps. Right now we're looking for active seeps. So we're looking for evidence of methane seep associated.
Oh, yeah, okay,
organisms.
so,
the organisms.
Chat,
that
I will
live,
pay attention to you in just a second,
that
but real
live there
quick.
because of the methane. Ancient seeps are seeps that were there in the past, and we can find the geological evidence for it.
Yeah,
So it's
look at the
usually
cukes.
certain rock formations. It's carbonate that is being deposited there because of the because of the organisms there eating all the methane. So we will only find the carbonates and some evidence for past life there. So sometimes you can find the clam shells if there were clam fields there. You can find fossil records of microbes as well. I saw a paper regarding that.
So turning that down a little. So these no, it went away. But yeah, that's a 3D map of the ocean floor that they made the night before. You can see Sebastian traveling along it. Have you seen the new billionaire submarine that was announced like 6 days ago?
That's again why one of the reasons why the seeps are so interesting.
Yeah. I don't know how I feel about that.
Project.
Let them go nuts. Either they contribute to science and exploration or Darwinism takes its course. I'm content with either. The issue with the Ocean Gate sub was that his big thing was like innovation and all these safety regulations get in the way of innovation.
Not possible.
This deep under the ocean, you might as well be in space. Those safety regulations are there for a reason. We say it on this channel from time to time. Those regulations are written in blood.
Quite the
Ohio Man is going to join the other two Ohio. No!
could be more recent, but
What is it? Why is it always Ohio? What happens in Ohio that you need to do your best to get as far as possible away from Ohio?
Probably more accurate.
Inactive, yeah.
The render of it makes it look like a super fancy stamp press.
So we were talking
Ah.
about fossils.
Yeah, we get
extinct
All underwater regulations and rules are written in blood, TBH. Yeah.
Yeah, I did some research yesterday and I couldn't really find a
Um.
term for
Oh,
it.
the ROV pilots are using game controllers, I think. We've seen in their control room before, and again, it's going to vary from ship to ship. I think we saw in the Nautilus, possibly the Okeanos control room, and they have like proprietary controllers with joysticks. But then we did also see the Xbox controller sitting right over on the side. It was great. I got really excited.
ridge on the map.
I guess this Ohio must be really boring, so everyone looks for something to make entries. Sent you pick for post-stream enjoyment. Thank you, Will. People don't realize safety laws are innovation. We didn't have them for thousands of years, and we were worse for it. Yeah. Industrial evolution and its consequences have been... Never mind. Is this a methane deposit like methane hydrate? Unsure? TK says Ohio is miles of corn and terrible cities. Trust me, I am an Ohio man. TK, I feel like this partly explains your lightly cryptid status. But yeah, I believe the methane seeps we are currently looking for, they've been talking about organic methane seeps, which is when material decomposes and gives off methane, and how those can be very short-lived. I believe we are looking for a geological methane seep, as we are in a subduction zone where one continental plate is sliding under another. So that's kind of what we're looking for here. Will Sleeps Not says tiny moving parts are hard to clean. Send in the children. Thanks, Snowpiercer. Yeah. Yeah. As much as Minecraft proves that children crave the mines, we should absolutely not put the children in the mines. Just remember, the airplane was invented by two Ohio men. Isn't it like, doesn't Ohio also supply the largest number of astronauts? What is going on in Ohio? The size was actually one factor. They used so many kids in mind. I wish that was a joke. No! Children yearn for the depths. Send the children to the bottom of the ocean. Just put them in the bottom of the ocean. I don't know why that's funny to me. That's really messed up. They crave diamonds. Mines have diamonds. Therefore, children love mines. Do they?
19.
First person to step on the moon was an Ohio man. Yeah, it's...
much closer together than they were before.
Oh,
we're kind of zigging and zagging here.
Yeah.
we might...
Checking out
Is it
geological
happening?
Features that are hopefully interesting and might harbor.
Oh, yeah.
methane
Astronauts,
seeds.
divers, presidents. Man, what the heck, Ohio?
At the very least, I feel like we're seeing a lot more life around these rocky outcrops than we are on these flatter bits. So
Yeah, absolutely. what do they call again? I have a Xeno Fire Force.
You know what the kids say? What do the kids say?
Fire Force.
Rizzler, Sigma,
Xeno Fire Force. That was a
Ohio?
very interesting anemone.
I guess
actually.
that is what the kids say. Ohio Riz Skibbity Go.
What are the things you science people do to prepare for an endure 24 hours?
Oh,
life like
what
this?
is what
Lots
is
of
that?
Coffee.
Yeah.
What else do we do?
Naps.
Naps. Yeah,
I
you
don't
work
stop
in
and get
shifts,
help.
Ideally.
Yeah.
Naps,
No.
juice boxes,
What
cookies.
is all that?
What is that
What
up
is
there?
that? Could we check that out?
Oh, what it
Ooh.
I don't have my little pointer thing.
is that a whale fall? Guys, hold on.
Oh.
I'm sure. Is it a whale fall? Is it a whale
What
fall?
That looks like that looks like a clam field.
Okay, no, clamfield. Which is
Oh
still
my
good.
God.
That means methane.
Oh, could it be?
We do want to stop here. Yes, absolutely. Oh my God.
Can we stop?
So this is what we've been looking for.
Yes, looks
This
like-
indicates
Oh
methane.
my God! I think we did it. I think we might have done it.
Ah,
Maybe.
Good Lord.
imagine the smell. I will not. Apparently, it's awful.
Woohoo!
Yay!
Okay, the sampling starts.
Let's go. Puchy, puchy, puchy. Wow.
Yeah, so those were the types of mussels we were looking for.
Oh my
Yes,
God, that's
yes.
wonderful.
Those are metal
It's
Yes.
a
Yeah, it's sea pig
get
in the
Alex?
corner.
yes.
Look at that boy.
the giant clams also.
Hey, those are the nothing seeps.
Oh my God.
Sebastian needs an
wow.
Oh,
Yes.
God. And they're alive. They are alive. Ah. Time to science.
Oh, okay, let me just take some.
Oh yeah.
Oh gosh, they are pretty. Scale is hard to tell. But those are pretty big, I think.
Can you
So
send me
getting
that
some-
picture
some
you,
people-
you took?
Yes.
I'm not at a
of
good
course.
angle to take any from where I'm sitting.
I'm not at a great angle either, but I'm gonna wait.
We can
Send
take
it,
lots
and,
and
we have a
lots
group
of
chat.
shots from the
So,
camera. But,
I'm gonna mark this as an observation.
yeah, I think that would be a good idea.
Absolutely.
Underwater methane is literally 100k
Oh.
or 10k.
Yeah, some of our
old
collaborators
farts,
already
yeah.
congratulated us, so they were watching the live stream.
Reggie says those are probably a foot long or
This
something.
is amazing.
So, and
Oh, there.
so this is one of the things we said was that
Yes.
there may not be obvious
Let's go!
puzzles.
Yeah, I don't see any bubbles on the sonar.
No.
No?
Nothing that seems like
Yeah, hard to say without the lasers.
super standouty. Wow. look
Ocean methane exists. Smit ocean. Ah! Yeah.
Well, it just could be a very small one.
Yeah.
It doesn't necessarily have to be
Oh.
extensive, but wow.
Crab.
Seen
Crab.
bubbles?
Crab. Crab.
Maybe they're just like super small.
Wait, you see the bubbles?
There might be small amounts on the.
They have better cameras than we do. They might be seeing a different.
Oh wow.
That's so
wow.
sharp.
Oh my God.
What an incredible. There they are.
That's,
Yeah.
That's
Oh,
amazing.
they're bubbles.
They're just they're
They're just
teeny.
very, very
They're just
tiny.
very, very
Because the
teeny.
ground is so packed. in,
Yeah.
Probably.
Oh my God. This is amazing.
Oh, there.
Wow.
Wow. Yeah,
Nice
we
to
just
watch them celebrate.
stumbled
Yeah.
right over it.
Okay. yeah.
Literally.
Yes, small, like the quill children.
Oh my God.
Oh, look over there. Wow.
Let's go.
Can we pan up a little bit too?
Yeah, is there more? I mean,
I
there-
think there's more.
Yeah, we
Up,
should
there more?
up the mountain
definitely
a little bit.
check this whole area out.
Yeah, yeah,
Oh
yeah.
yeah. We will be spending some time here.
Oh
Cool, it's a great hole. There are many who don't like it. This one is ours, yes.
yeah, I mean, we'll sample here and then we'll just keep going.
It's a pixel, one little pixel of bubble.
Water before we start doing cores.
Oh, so they're gonna take soil push cores.
Oh yes.
We'll take some clamps for you. Oh my gosh.
Do we want to consider water first, then cores, so we
Yeah,
don't disrupt
probably.
the sediment?
Yeah.
Oh, that's a good
probably
point. Yeah,
idea.
water and then cores.
With the Niskin first, and then. we
I
can
guess
start
the
to-
question is how many Niskins? Since we found one and we were all looking for it, do we want to fire all of them and just get
Oh,
the
gosh,
water
no.
through what we can?
That's
So
a
we're
good
gonna start with putting the temperature
Yeah.
probe in there.
Yeah,
Adam
that's
was,
a good
was just
idea.
asking if we should.
Yeah.
Okay,
do that.
so temperature probe. Guys, watch. Oh no.
And now we don't have the
Watch
oxygen sensor on the
here.
arm anymore.
Watch here.
We don't.
No, I think it's back behind the camera.
Oh no.
Yeah,
Oh, you need to grab it. Oh.
that's all right.
You can check
good.
the. Yeah, I'll check the VOD later.
Oh my god.
Let's see.
Oh man, I'm feeling really emotional right now.
Yeah.
I'm getting a little choked up. Oh, this was a long time.
This is so
coming.
cool. This is so cool.
This is a lot of work.
Wow. Months and months of looking at data.
Yes, and looking at
Scientists
the
are very
bathymetry and
easily amused.
stressing and going, are we sure we're going in the right place?
But also, this is like a
Find
huge W.
anything? Oh, this is so cool. This is so cool.
Party in the controller.
Party on the controller.
Let's go, Marty. You can do it.
So the temperature probe is going in. The ambient temperature was 1.4.
So for those of you who are just joining us right now, we have the mission control room. We are going to take some pictures. We've been in the water since about 5 a.m. this morning.
Imagine spending months of your life trying to find something all live, then find it, and everyone doesn't understand because wait, what?
12 hours.
That's it?
Wow.
Okay, so they're doing the temperature probe now. Yeah, it doesn't. It's not like anything super flashy.
Yes. Teamwork makes the dream work.
And it's a shot in the dark, trying to find this kind of ecosystem.
Oh, there's someone in the back like doing a talk. I just searched the oceans for months. I dare say, you're hard to please.
No.
Yeah, we're not seeing dramatic. I was expecting a very dramatic spike like we see at the hydrothermal vents. But there's not. We're not really seeing that. It went up like 1/100th of a degree. Right? A 10th of a degree? Look, I'm stupid, chat. Yeah, there's no change. I was expecting that to spike dramatically. That's honestly kind of weird. That's weird, right?
So this is the deepest methane seep site that was found off of Chile,
Let's go.
and the northernmost. That's what we just heard from a collaborator.
Deepest and northernmost
That's
of the coast
amazing.
of Chile.
Chile, according to Javier. So? Not only did we find one, we found this one.
So we are at 2,835 meters.
In terms of sampling, I think since we have
And-
one, I think we should. But right here, I would say we should fire all the Niskins and get all the water before we start.
Oh, they are. They're gonna do it. They're firing all the Deniskins.
And then if
0.01 degrees. Oh my gosh, it's amazing. Usually,
Just here.
at least with the hydrothermal vents, it's... I mean, to put it this way, quite a few things got melted trying to sample off those hydrothermal vents.
All right. Alex is having a field day.
amazing. You're amazing. I gotta catch my breath. I'm all verklempt.
Cool.
They're just losing it in the background. This is great. Ah.
my gosh. This is so cool. Hang on, I gotta touch my husband and
So all we just do is first grab the water before we kick up sediment, and then everybody, I mean, basically, we're just gonna, everybody's push course. That's true. There's a lot there.
So those fire miskins which collect water samples. Man, that's what I'm saying. Keep at least one in reserve.
As many as you can. Yes. Oh, the cores all around. Samples on me.
Course all around samples on me. Yes, those are the clams that indicate a methane seep. It's cute to see smart people happy. Yee.
5 liters for sure, and we can keep 2 of the small ones.
Probably. I mean,
They are. Like they were saying, this is a lot of oh my gosh, yawn. This is a lot of work coming to fruition.
I'm very happy for very happy. We know very good temperature variations and bubbles, and then you have your niskins, and you can always come back to it. Now we know where I need to know. You make an idea of we're here now. You can. So,
one idea is to put down a marker. One idea is to put down a marker and then come back.
I think they're still trying to figure out what to do.
Because they're not understanding why we all are happy.
Indeed, this is like looking for a needle stack the size of Ohio or something. Yeah.
For days, we've been looking at the bathymetry data and planning this for months, and we weren't sure if we were going to be able to find one this deep. And not only did we find one, we found the deepest one found off of Chile. So this is incredible. We're going to sample the heck out of it, get sediment cores, get water,
And are there more deeper than this in the world?
I don't know about worldwide, but off of Chile.
Worldwide, there are some deeper.
20 years ago,
How many
discovered the first methane seep
So
in
this
Chile.
was
20
the
years.
20 years ago, they discovered the first methane seep off of Chile.
the question
So this
from
is-
us is, do we want to put a marker here and not sample right away and explore? No, but look around this area. No, it's fair, because if we fire the bottles now,
That's true.
what if we find something bigger right here, right? So,
Mm.
so that we're not talking about.
Sorry,
like,
I was
moving
speaking at
around.
the. I was speaking. Brian, hello.
Yeah, we have
What?
plenty of time.
I
Or
guess
Brandon, sorry.
we could
Hello.
Make it quick. I mean,
We can also
yeah.
make it for the next couple of hours.
We've got plenty of time. It's not going to go anywhere.
That's
Just realized that I missed. They found a methane seep. They found a methane seep.
15 minutes to just peruse
Another periphery.
and see,
Yeah,
right,
let's
if there's
look
any
around
more
and
in this
see.
area.
Maybe there's more. Maybe it's more extensive than we
Let's
think.
consider, imagine that for whatever reason the rubber fence has to go up. Yeah, so like here. Let's consider if whatever reason there's something in the rubber fence. we
Yeah,
have to
I think-
leave.
Yeah, because
So,
if
just a
we
little
fill
around.
up,
Just
if
a
we
little.
fill up with samples, we have to go up to the surface. So
That was the
this
suggestion,
spot.
is we mark it,
yeah.
look around a little bit,
Look
and
around,
see
maybe
if,
we'll find
you know,
more. But this is, for those of you in the chat, this is incredibly exciting. This is an awesome source of energy for microbes and other life down here. This is all life from chemistry. This is a place in the dark. There's no light from energy from light, so they're feeding off of this chemical energy. So this is incredible. And we could learn a lot about how life in the dark can exist. So,
we have 15 minutes while it's still our show. Okay.
Also, please, y'all, don't forget it's dinner time. I know it's exciting.
Sample bottle now. Explore sample. Yeah. That's what I'm saying. Like, I get that they found it and they wanna
I'm
collect right now,
over on
but
dive watch. I was sitting in the background, just lurking in the shadows, but
all right. But yeah, these clams indicate a methane seep.
oh my goodness.
Wow.
Well, I've got about 15 minutes left, so I'm gonna sit down and I'm gonna
Yes. Yes. Wow, look at that. If you zoom in really close, you can see little bubbles of methane escaping from underground. They're so tiny, we would never have picked them up on our sonar.
Here's to sample before you can contaminate. That site has to be
Thanks, Jeff.
huge.
Hi.
Yeah. I want to peek over at their chat.
Yeah, wow. So we may find more of these seats on this particular
on their
ridge if it's a
chat
good
says,
spot
"Woo!
for
Now
it."
you get to have a stinky
So
lab."
we're gonna
Yes.
put a little marker down, take some pictures here, and then I think we'll do a little bit more exploring before we start filling up the ROV with samples. But we can always fill it up and come back for more. This is incredible. This is super amazing. Wow. I'm trying to get my heart rate to slow down. I'm so excited. Wow.
Ah, squat lobster.
Yeah, look at that.
So beautiful.
Oh, stinky lab. That's the smell of science. It is.
Lots of life here.
No, I saw it and I said that I didn't think it was important.
They want
Lots
to take
of
samples of the
bacterial
methane itself or the
production
earth around the methane to see what
Yeah,
lived
could
there.
be some
I
fecal
think,
casts as
I
well.
think, like both.
We've got these bivalves. We've got these amazing crustaceans here.
But
all
primarily
feeding
of
off
the
this
sea, as
energy,
they're looking for micro bacteria. There are
living out
two.
their lives in the dark, feeding off of geochemical energy from under the seafloor.
There are two astrobiologists
This was a long time
on the
coming.
expedition.
We spent countless hours looking at the data, mapping the seafloor, trying to figure out where the best possible places would be to find these, looking at fault lines, looking at ridges, and we just stumbled across this one along the path that we chose. We're so lucky. We are so fortunate right now. And you're all really fortunate to be sharing it with us. I'm so glad that people are here watching this right now. This is incredible. I think these are squat lobsters. We've been seeing a lot of them on the way up here, but we didn't see any seeps. And so we were like, well, we keep seeing the lobsters because they're often associated with the seeps, but we didn't see any seeps, and now here they are. There's lots of other little critters here too that we can check out. Yeah.
Yeah.
Little sea star there. Some isopods. Oh my gosh. Wow. Wow, I'm just speechless. I'm so happy to be here right now and to be in this moment. We worked really, really hard for this. Yes, so there's most likely a biofilm. I think that there's probably some detritus covering it. There's some greenish color around, or maybe gray. It's kind of hard to tell. But we will definitely take tons of sediment cores here, and if we find other spots, we'll take them there too. Get some of that surface sediment. Yeah, that green teal. I'm not sure if it's actually green or if that's a trick of the light. Over on the left there, around that other mussel bed, could be a biofilm. Can we just take a quick look at the one over on the left there? I don't know where the pointer ran off to. And put the light on that. I can't tell if there's a biofilm over there or if it's just shadow. Oh yeah. Yes.
If we leave here, don't we want to throw a lot of sediment?
Will it be okay if we lift up? Will we disturb it?
Oh,
I think we can be. Yeah, we can just kind of very slowly and gently back away. But we have a marker down, so we know where
so
to
I
come
think
back.
they are gonna continue to explore.
So, but yeah, it would be great if we could zoom in on the clam bed on the left here, because it looks like there might be
So excited for the
biofilm
submersives
on the surface.
Possibly, yeah.
Hard to tell, or
Like,
if
there's
that's
gonna
changes
be-
in the sediment. Yeah.
This is a lot of information. Gonna say hi to the sea pig. Yes.
Yeah, it's a little difficult to tell if it's a biofilm or if it's just differences in the sediment. There's like some white, sort of powdery stuff.
Oh yeah, just like about in line with my hat.
That's a
that
good question.
powdery
Why are
kinda-
the squat lobsters here smaller than the bigger ones we've seen elsewhere? I don't know.
Oh, so the two green lasers, you can barely see them, are 10 centimeters apart. They might have said that. I've been lightly distracted.
Yes. Yeah, definitely. We will definitely. Can we take a note of that? That white area above the clams there. Zoom in on it. Take a picture. And after we fly around a little bit, we can come back and take a close.
You wanted to stop and look at this one though, right?
Yeah, if we can just see. Yeah, if we can just zoom in on that for a second. Whatever works better for you guys.
There's just some stuff underneath this, so if we're concerned about these shells and things directly below.
I wonder what they do to calibrate the
Why
distance
don't we
of the
just-
laser pointers.
Can you just zoom in?
Oh, I never thought about that.
Yeah, that's fine. That'd be perfect.
Because it
there's
is
a sea
expected to be a fairly accurate tool.
Yeah.
I imagine there is a calibration process. Look at this sea pig! Look at that boy!
It looks like a biofilm, that white.
Oh yeah,
Yeah.
the white, there. Up
Why would there be marks in the sediment between the two clam colonies? Could be animals that were scooting around in between. Gorgeous. Wow. Oh my goodness.
Squeakers are squeaking. I'm gonna put the quail on
Yeah,
the screen.
I think.
There we go, stinky babies.
Yeah, hiking trails. Yeah, I think some of that
I think that's an excellent
those
choice for
changes in
underwater
coloration
research.
might be
Lower
microbial
fraction water,
mats.
digital cameras are very receptive to that wavelength.
We'll know better, of course, once we've sampled them, and then we can do some sequencing.
Yeah, I guess like red is the first color you lose at depth.
What do you want? You want an observation of it?
Now that I'm awake.
Thank you.
Can we get a close up of the white?
No, they were.
Oh yeah,
they
close
were
up with the
alive.
white sort of powdery substance there.
So talking with Nick, probably this is an
Oh.
inactive evidence is because you're going to see most
I
of
think-
the remnants of two
Did I stream Friday?
highly
But yeah, this is the first time they're all like healthy and running around, being really active. On the other hand, red is also the color that doesn't scare the fishies. Ah!
going down, but it was
Standard digital sensors have 2 green
We
pixels.
saw a couple bubbles coming out, so.
yeah, but
Two green pixels for one
Yeah,
red
there's a
and
few.
one
There's
pixel.
a few.
that-
Oh, okay, okay.
there.
Yeah.
So maybe it was active, I don't know.
Maybe it was more
Month
active
ago?
before.
Right?
Hard to say.
Months ago, maybe.
Maybe we'll find ones that are more active as we
Yeah,
keep
we'll look
looking.
at
Yeah.
Yeah.
This could be a seep on the decline. It's on the smaller side. We are seeing little bubbling out of here, but it's very, very small. That's, yeah.
So,
Oh, that is very exciting for a partner.
Oh wow, that's
Look at that.
amazing.
yeah.
Wow. It looks like cobwebs.
Would the babies try to eat the squatties?
Yeah.
Yes, they would, yes.
Oh gosh, no, not on site. Certainly, we would expect to see anaerobic methanotrophs at a site like this. Those are generally very common at methane seeps. There could also be aerobic methanotrophs, so microbes that feed off of methane. And there are a couple of different clades of those. Yeah. That I don't know. I mean, generally when you see any kind of bacterial mat, what holds that together would be what's called an exopolymeric substance. Or exopolysaccharide. So these are, it's literally like a goo that bacteria secrete to help them all stick together and form structure.
Bacterial
so these
mat
are
sounds a lot
big,
better than
sugar
bacterial
molecules,
goo.
basically, that can help bacteria stick together and form
Sugary
structures like
bacterial
mats.
goo.
I've seen microbial mats that even have like almost like a tower-like appearance. They grow upwards, not just outwards. So depends on like how dense the growth is.
The cobweb structure on that is kind of different than what I've seen in the past. I haven't seen one like that.
It's possible that it's methane hydrate. I don't know. Either way, we need to take a sample of that.
Do we want to decide that not to look around and just grab stuff here?
I think that we should look around a little bit more, just to see if we find something. Like there could be some more extensive seepage, just outside of our field of view right now. But I think we should definitely mark it, plan to come back. We have so many hours for this dive planned out, and we can just sample until we're ready to come out. So.
Oh, I'm dropping frames. Why am I dropping frames? I might have to end and restart just
the grad
quickly.
student takeover is coming up?
Yeah, 6 o'clock.
I think I got lucky. This is an exciting time for them to be taking over.
Yeah.
I know.
All right, well,
Hello, how are you? I'm good. I'm even better now, so now I can go to dinner and be like woohoo. So I have already messaged my wife and kids.
Victory dinner.
Because at 7 o'clock this morning, I was like, I really hope we find a mat today. And she texted me a couple times over the day, mat, and I'm like, no mat. Seep, no seep. But seep.
Yeah.
Sorry.
We
It's like
did
they're using a
it.
pair of... Oh, I wouldn't even know.
Why don't
I'm sure that
we,
information's available somewhere,
continue
but...
going along our planned path for a little bit, and just have a look about.
You want to do a search pattern? Down, up?
Yeah, maybe zigzag along this little area here, because we're right at the base of a little ridge here, so.
Hey, hello! Yeah, we're watching some fishies.
Sure,
Welcome.
yeah. Maybe we'll see something bigger. Maybe this is just kind of the outskirts.
You asking for too much.
Yeah, I'm gonna. So we're gonna get stuff in the bottom. So I can grab one and back to this.
So this is already a victory. We have already scored the W. But they are going to look around and see if they can't find anything else. Perhaps something a little more active. A little more recent. Since there was talk that this one has perhaps begun to decline.
Little rocky outcrop here.
That's interesting. That outcrop looks like there might be some interesting
We're
stuff
looking
on it,
at
so
methane
Oh, yeah.
I want to
seeps.
look at that too, probably.
Yeah, we could certainly zoom in on that.
They
So
just
this
found
is
the
animal
deepest
sample.
and
The
northernmost
depression
methane
that
seep
we're
off
seeing
the
in all the lines
coast
on
of
there,
Chile.
Is that kind
Obviously,
of
the
a
planes
secondary
being
sign of
equidistant in every distance from the ROV.
I think generally
means
what,
that the lasers need
like,
to be perfectly parallel.
physical
Yeah,
marks
yeah.
of the seep would be like pock marks.
Okay.
Those could be
So we're
animal
looking-
tracks, maybe.
We're looking for clams because clams indicate methane seeps. there are 2 astrobiologists aboard this expedition.
So, methane can be used by microbes as a source of carbon.
Yes,
and as
yes.
a source of energy. And there's a couple of different pathways that they can use for this.
Do they know its origin?
But it's basically a chemical form of primary productivity. So rather than using light and carbon dioxide, using methane.
Yeah.
And so this is a small example of a seep. There are certainly larger
My
ones
goodness,
out there.
that is a
are
good
more actively
question.
Bubbling
And
that
how
you may
things
stumble
get
upon.
to vents and seeps is definitely something they're still
This
studying.
is a great example
But
of
I think I'm actually going to
geochemistry fueling life, which is super exciting.
I'm gonna see if someone has asked that in their chat, and if they haven't.
So, before we do a ton of sampling here, we're just going to take a quick look around. We kind of zigzag around this path that we had started on and see if we can find anything else as we go.
You guys want to take a look at this formation over here?
Where's that at?
It's dead ahead.
Yeah, I can't quite.
There you
okay,
go.
yeah.
Apologies if this has
Sure,
been
we can head
answered,
in
but
that
how do
general-
clams
that general direction?
get to the seeps?
Yeah, so we expect that if we find life on another world in our solar system, like Titan
So
or
this is big
Enceladus
because
or Europa, that life will be fueled by chemical energy, because those worlds are much further from the sun, covered with ice, and the life may be under meters and meters of ice.
you
And
question
so
of abundance
chemical
of
energy
resources.
probably
How do they
supplies
get
energy
there?
necessary for
Because
life.
things are so barren.
How do the clams find or get to the seeps?
There you
Oh,
go. That's your question.
Do not know very much about bivalve dispersal, because I don't study mollusks. I know that their larvae disperse and are transported by water, but
I don't know much about bivalves, but if you look up the concept of
We have,
yeah, let's go ahead up there to that white. Sorry, Soph, go ahead.
I think Nick has something he might want to say about the bivalves.
Oh yeah, Nick, please.
Let's go. This is a good question.
Yeah, we have a bivalve expert here.
Mecodius.
Yeah, well, bivalves have a veliger larva, which has a planktonic stage. It really depends on it's species specific, and usually they reach a stage where they search for chemical cues to be able to settle down.
Is there
and
some
usually
more?
they like to settle down
There's
where
some
the
more.
adults are. So, in a way, the adults release chemical cues for the larvae.
Oh
So that's-
wow!
Yeah, I think there's
So
more
they
seeps.
they
There's
have
some
like a
behind my
chemical
head.
language. That's very cool. Bacteria have a chemical language too, so they have something in common.
Some of the cues for those larvae, actually, there's some evidence that they might be quorum sensing related in some way, like some of the
Can
stuff
you
that Aparna and I work on.
Zoom in on some of that white material. I think that might
There's
be
even
another
more.
mat there.
So yeah, that was an excellent question.
Definitely more of those clams, which is a great sign.
There's a lot of it. This
Yeah,
spot.
yeah. All right, we'll have to think about, maybe put a marker down here. We'll have to think about how many cores we can take. Depending on how extensive this is, this might be a multiple dive. Yeah, you zoom in there when we'll make a note of that in the sea log.
Ah, okay. So there are biologically
know.
driven
Pictures here from the
seeps,
outside.
and
That
then
to me
like
looks more like a biofilm than ice.
geologically driven seeps. I'm actually
good to me.
not sure which one we're looking at. I thought we were looking for geological because
Could
we're
be
in a
vent and
subduction
decline,
zone
'cause it is kind of small.
where the plates are meeting, but perhaps not.
And there's some dead clams here.
Can we zoom in on the worm, please?
We can hear
Oh
the
yes,
playback
can we
of
zoom in
Schmidt
on the
Ocean
worm,
Stream.
please, for Nick? Long boy. It's pretty.
Do you mean the scientists? I just need to stop talking over them, that's all.
Okay, yeah, we can make a note of that. Seek 2, Aparna would like a sample from.
You should not be hearing
Put on the back of a sheet paper as we're starting to take notes.
echoes from
And then
me.
once we've had a pretty good lay of the land, we can figure out what's going where. Yeah, I see that spot up ahead. We can head over there next.
So, we have hit 6 o'clock. So,
No
we
echoes?
want to
Okay.
let the grad students take over for a little bit and share in this discovery.
I think that would be very exciting.
I am gonna go upstairs and force myself to have dinner. I hopefully can put the food in my mouth while watching the TV and not
my
hit myself in the
goodness.
face with the fork.
Yeah.
Queen, you
Of course,
have
we
a
find
plate
it right at
Schmidt
dinner
Ocean
time.
stream in your own stream.
So-
Wait, no,
We were
I read
asking
that.
the chef to put aside a
No,
plate for
echoes?
me.
Okay.
I'm not leaving this room.
Oh, sorry.
Bandana fell off. I'm so excited.
Well,
Yeah, I just need to stop talking over them. Imagine how many dead dinosaurs must be buried here for there to still release fart gas after all those millennia, right? Very robust ocean farts.
Good call. I like it.
okay. Like things that they won't put the
Release the grads.
In a turn of events, the crew
Yes,
are
I'm
now
looking
putting
some of the
highest
comments
rated-
from the viewers, and it looks so far it looks like this is a vent in decline. However, still this is an amazing discovery of the day.
Very happy.
So
And we may find something more active further ahead. These kinds of seeps, as we said earlier, are transient. They may not last for very long. They come and go, so we may find more active ones later. We've got a lot of area that we could still potentially cover, so
Yeah.
the night is young. We had 10 more hours planned for this dive, so
Oh gosh.
we're certainly in no rush.
cucumber.
Sea cucumber, he's doing that little
Oh.
the Tina Belcher dance. I don't know how else to describe that. Looks like there's another white mat over there. Yeah, that one up ahead, Aparna. Yeah,
Do a little
Aparna's
dance.
gonna have a field day here. She's all about microbial mats. So yeah, it looks like there's some more along the other side there.
Check out this one.
Yeah,
You wanna sneak down and check those out?
sure.
Headless chicken monster? No, not headless chicken monster. What does it mean?
Another indication that this may be a seep on decline is I see normal animals that I would see in any other part, like within meters. Again, people should appreciate the ephemeral nature of these ecosystems.
He's had a
Look
big
at
couple
the
of days.
giggles. The delighted scientist noises. Ah! Headless chicken monster is a type of sea cucumber that swims and looks like.
We don't have a
Really?
thermal plot of this area. These seeps seem the one that we did measure the temperature on seems somewhat cold, so that would be another indicator maybe that it's in decline. But it's still interesting. Yeah, look at all that material there.
We are not in Mexico. We are in northern Chile, of the coast of Atacama Desert.
That question was directed to Jess earlier because she lives.
okay.
Yeah, she lives off of Mexico on her boat. Yeah, no, that was from an earlier question. But yes, we are off the coast of the Atacama Desert in Chile, and we found what we believe are evidence of the deepest yet methane seeps located off the coast of Chile. That is super cool. Look at all that fluffy,
It's so
feathery.
fluffy.
Yeah.
So that's a bacterial
And
matting
there's like some gray material and then the white.
that's present in methane seeps. For
They
those
have
that
been
are
more active in the past.
Let me turn this down for just a
The
minute.
color variation may be different species in bacteria, possibly, or also differences in the sediment.
So we are with Sebastian, the unmanned ROV, who is off the research vessel, the Falcor 2, diving the Atacama Trench at 2,800 meters deep. Oh gosh, and this is an exciting expedition. Basically, there are 2 astrobiologists on the ship, and they were looking for these methane seeps because this might inform how life would exist on other planets. Mars keeps getting mentioned. These astrobiologists, I believe, were previously in the Atacama Desert, also looking at things there. So a lot of this is a very microbial-driven expedition. We're looking for microbes, not so much larger animals. Although, we are looking for clams because those indicate these methane seeps. This is a really cool expedition. I am hype. I am hype for these stinky ocean farts. Let's go. You know what that is? Wasn't unmanned surface, unmanned ID surface immediately? Huh? The event, Sebastian. I was just thinking about you. A friend of mine who's a diver just finished his full 3D model of a sunken ship in Lake Washington. That's so cool. Yeah, there's ocean exploration is so hecking neat. And I'm so hyped. I'm so hyped that it's tying into space exploration. This is the best of all worlds. Never mind, just listen to the clip. There's another scientist in the background talking about bivalve dispersal in exactly the same way another scientist on the mic said it. Ah, okay. Yeah, I feel bad talking over the scientists. I try not to do it so much. Like if I want to address chat, I try to turn them down.
Keep looking until we find them. Look at all this.
But they also talk over each other, so he's scary good at what he does. His day job is training underwater. Oh gosh, so he's the boss.
Yeah.
Like underwater, you got like divers, and then you've got like underwater welders, and you've got the guy that trains underwater. Well, he's the end boss. He's the big boss that you have to fight before you fight Cthulhu. I don't know what I'm saying. How are you getting too excited about the ocean to listen to ocean facts? It's true!
Yeah.
Oh. That's a commercial dive. Oh, the babies are squeaking. Let me turn the baby cam back on.
Oh,
while
were
we
you
zoom
here the
around.
other day when I was narrating as Linda Belcher?
Wait, what?
Or was that just a strange coincidence?
It's a commercial diver, which is
at
completely
that
different beast from what I
go.
do in my free time. Yeah.
All right, Bobby. Oh, wow.
There's some good wording from the audience there.
Yeah.
Precipice of an ancient ship.
Yeah,
that's pretty nice.
See, it's
beautiful.
Cthulhu.
It's
I can
poetry
comprehend
right
these
there.
words just fine. Skill issue.
Oh boy, a brachiopod.
He's 5'8". His father was Dutch, so it makes this right.
I'm so glad y'all are here for this.
One of the lab staff went to a community college in like this
scoot ahead and see
6'8".
what's
Sorry,
over this
6'8". I don't know. I don't know
little
numbers.
here.
He's 6'8". His father was Dutch, so it makes sense. I know a lab staff who went to college in just kind of a relatively rural area, and there was
We could
a
definitely sample here, but
kind of historic
more, I bet.
settlement of Europeans. So they would just be wandering around, and then there would just be like some dude who's like 7 feet for no reason, just going to
Wow.
class. Like, where did you come from?
it goes pretty far.
Just a bunch of spheres. I don't get the confusion. But yeah, this. Oh gosh, look at all of this.
Yeah, this looks mostly inactive.
We found this Ukrainian. It's no.
couldn't say
I
how
think it actually was. I think
recent.
it actually was a Ukrainian. Yeah. First girlfriend was Ukrainian. She was 6'2". Was so tall.
Few clams here still, and some growth, some worms. Sticking his tongue out at us. Yeah, let's keep going. No, I don't think we're gonna go any deeper today. We're gonna climb a little higher, if anything. Right now we're at about 2,800 meters, a little more than that. It's pretty deep for a methane seep.
Haya's autonomous
Some
form
more.
confirmed for botlet. Look, okay. First of all, look at
Yeah,
that.
can we
Look
get
at-
a zoom in on?
Look at that.
There's definitely worms.
These are calcium,
Oh.
calcium carbonate polychaete tubes.
Polychaete tubes.
Polychaete tubes?
Some that may be still alive.
Okay. Look. Haya's autonomous form confirmed for botlet. I am 5'4". Canonically. If you don't count the 2-story servers that I'm being hosted on, which, depending on your source, is the exact same height as Vegeta. Chat, I'm not short. I'm saying Prince height, okay? Somehow still taller than Spicy. Yes, I'm like 1 centimeter taller than Spicy. Meaning I tower over Spicy.
Husband got my all caps.
Circle
text
worms,
from
if I'm not
from a
mistaken.
moment
Yeah.
ago. He's probably putting the baby down for a nap.
Hi, is Vegeta. Got it. Yes.
You're like, what did I miss?
Size of Vegeta or size of Vegeta's ego? I'm the size of Vegeta, and the servers I'm hosted on are the size of Vegeta's ego.
Okay, sorry.
You were
Oh,
all
shrimp?
small.
Okay, never mind. Well, no, look,
Oh,
check it
yeah,
out. Check it
shrimp.
Those are alive. Okay, no, those are alive.
out. Wow, look at all those guys. And their little feathery bits.
Oh, those are. Wait, those worms are alive. I thought they were fossils.
Polychaete field or something?
Yeah.
Shrimp.
Yeah.
Worms.
Take a picture, make a note of this one. This is very, very cool.
Yeah, those feathery things. Those are the worms coming out of the tubes.
Yeah.
Cerlupid polychaete worms.
My dad
Okay.
just texted me. He's like, "Are you still on the live stream?" I was like, "Yeah."
Yeah, Nosium, thank you so much for coming in. Let me shout out
Polychaete
Nosium.
fields forever. I like it.
It's always good to see you in here. I hope you are doing well and enjoy your day. There we go.
Wow.
Guys, go check out Nosium.
Polychaete is Polychaete.
C-H-E-T, yeah.
Whatever you want.
Polychaete
Typically called
worms.
keel
yeah.
worms. Though that is a specific type. Got it.
Tube worms.
Unheard is amazing. I will check it out. That's high. That's high
And we
praise
see
from
some
you.
Yes. former ones over here
Oh,
too as well.
oh, okay.
The former shells.
Sound-based detective game. I will check that out.
And these clams.
I'm actually going to write this down because it's well established that I have a 30-second memory and I'm going to forget this the moment I hit stop streaming. I'm half deaf and even I beat it. Ah.
I don't know why. We'll definitely come back and get plenty of samples from here. I'm also hoping to study the microbiome of some of these animals, so that'll be super cool. Are they static? The worms? Yeah, I think they form these like solid casings, and then their mouth parts come out and move. Can we go out and further to the right?
All these animals are benthic, so at the adult stage they cannot move. However, they can move at the larval stage.
Yep.
They can disperse, not
Right.
really move.
Right.
Yeah, yeah,
right.
yeah.
They're dispersed in the currents, right? Yeah. Here? Oh, can we zoom in on this thing here? Sure.
I like oceanographic scientists because goldfish, yeah.
As
That's
they
where my
grow,
memories
they secrete calcium carbonate and they make those tubes. Correct.
That's so cool. Life making rocks. Rocks making life. That's what we're all about here. Oh.
Too much excitement there.
Too much excitement.
My grandmother would have said, I dropped my watch. She made a really loud
We
noise.
forgot how to walk when they're adults. Yeah,
She's
yeah.
watching this somewhere. She'd be so proud. She'd be like, Lauren discovered a... What do you call it? A seek. A seep. Maybe some biofilm material down there.
Oh gosh, so that's the 3D
The green thing
representation
might be
of
another one of those sea cucumbers.
that's where we're going.
Yeah.
We will in fact need eyes. We are searching for visual signs of methane.
Going to the right? Was the direction we were going? It feels like we're kind of circling this spot, but that's probably good.
Yeah, to stay in this area. So again, we're looking for these clams. These white
This here looks like another clam, but
yeah. Because they indicate the
some
seeps.
kind of microbial mat next to it. Thank you. Alright, why don't we, want to keep going? Yeah, let's keep circling around this spot, and then maybe we can start doing a ziggity zag.
Clams are probably about 10 to 20 centimeters instead of a foot long. Oh yeah, as the lasers show. They're smaller than I thought they were.
Probably the bacteria, because they're kind of the base of the food web.
Still decent size though.
Larva, I would
Oh
think.
my gosh, yawn,
For
sorry.
the sea cucumber question, if we look around, I really think we're gonna find the same species we're finding in other parts. It's just
Yeah,
we're focusing on the
Yeah, we've seen a
one
few of
species
these
we see.
here and there. Yeah. This looks like more. There's a few live clams, but I think there's a lot of dead ones here too. Yeah, I think we can maybe kind of.
So they mentioned this might be a seep in decline. It's always weirdly somber.
The
way
acorn
things-
worm could be used as an indication that the benthic environment is, let's say, back to normal after the.
Oh yeah, kind of a transition away from this type of ecosystem.
Yeah, so it's sort of this area being in decline. The amount of life is going to reduce.
On the other hand, if these are
It's
in
oddly
decline,
sad.
we'll probably see more carbonates potentially.
Oh,
So
maybe not in decline.
tough to say.
Today I learned the Rubicon River
Yeah,
has a
yeah.
teeny water course
Yeah,
on the
can go up
east
a little bit
coast.
higher. Get more of a wide-angle shot.
I learned the Rubicon River has a tiny watercourse on the east coast of Italy, just north of the Principality of San Marino. This completely overturned my understanding of certain historic events. Brick, explain for the class. But explain with crayons, because we're stupid. By we're I mean me. I'm stupid. I need to head off now. Yeah, Brick. Thank you so much for coming in and hanging out. This was so cool. Please have a good day.
Higher in
Have an excellent day. Let's go.
more shallow. Yeah.
You have been amazing to see this area at its peak activity.
Yeah. At its peak, it would have been incredible.
It does make me think that perhaps we'll find more.
Where are we? We are in the. Oh,
activity
wait. Let me.
close by.
Because this is actually really cool, Brick. We are in the. Oh, Rev. Hello. It's my. Oh no! Happy raid. Oh gosh, that's a lot of people. Hi, welcome raiders. Wait, no, guy. Wait, no. Wells welcome! Welcome, Raiders. Let me turn this down. Let me turn this down real quick. Hi, Rev the 10th. Let me get a shout out going. It's your birthday. Happy birthday.
I was talking to my colleagues there.
Uh.
And apparently the life expectancy
Hello.
of the shallow water ecosystem, for the people that know, that was next to the
Jalpius?
north.
Corvus the Greedy? I'm gonna mispronounce everybody's name. Liar. Liar sighted. Hello.
They were measuring
Wells Moore?
decades.
Scumbag Sorcerer? Hunting Laughter? Hello, how is Elden Ring?
Yeah.
Hey yo! Happy birthday to you. Or wait, no. Happy birthday to you. Happy birthday to you. Happy birthday, dear Wells Moore. Happy birthday to you. So welcome, Raiders. Hiya, my name is Hyacinth. I am a flower-themed AI. I stream horror games, cozy games, and real-life ocean exploration. We are currently doing a watch-along with an organization called Schmidt Ocean. They are doing a lot of underwater mapping the ocean floor because we don't know a whole lot about it, but they also do live dives. We are watching feed from the ROV Sub-Bastion, launched off of the research vessel, the Falcor 2, because scientists are nerds. And we just had a major discovery. They have discovered a methane seep, the deepest and northernmost methane seep off the coast of Chile. Moa is getting a lot better at Elden Ring. Let's go! I'm loving the fish. What is... Oh gosh, I missed it. My apologies. But yeah, so this is a particularly interesting expedition. There are 2 astrobiologists, like Astro is in space. Astrobiologists on this expedition, and they are looking at the they looked at the conditions in the Atacama Desert, and we are now in the Atacama Trench. They're taking microbe samples, and they're kind of using this data to possibly form theories about how life might exist on Mars. So this is really looking forward into the future. There will also be a Q&A on NASA's channel tomorrow with these 2 astrobiologists. It's super cool. There are very intelligent people in chat who keep me from telling too many lies on the internet. I don't actually know that much about it. I'm just really excited about this expedition. But yeah, live dives are very neat. Like I said, this is happening live. Let me get chat let me do a shout out for Schmidt Ocean as well. But thank you, everyone. I will turn the volume back up in a minute so we can hear the scientists. Like I said, we found the methane seep. Mission accomplished. We did it. Breath the Temp, thank you for the follow. And Scumbag Sorcerer, thank you for the follow. Let's go. This is their YouTube channel. They do have a very active open YouTube chat, which a lot of these dive streams don't have an open YouTube chat. If you go there, behave yourself. Methane seep plus astrobiologist, Mars methane science. Also also Europa and Enceladus. I believe they are kind of looking ahead of the ducks to Europa and Enceladus. I have approximate knowledge of a great many things. It's true. Everyone new here. No! I'm gonna catch the ducks. I'm gonna catch the... I got one! I got one! I got one! It went out! Come back! No! Not the ducks! Not the ducks! No! Metaphysical engineer, thank you for the follow! Hello! Oh, let's turn on the volume because this...
Travel.
What are you? Are they gonna talk about the fish? But yeah, welcome everyone. Hello.
I'm just saying, hey, pull over and look at that thing over there.
Adhob? I'm sorry, I'm probably mispronouncing your name. But yeah, so that 3D map up there,
We are in a subduction
they mapped
zone
last
right now,
night,
so we are on
and
the
now they're
South
following
American
that
plate.
map.
The Nazca plate slides under this one. So we're on the South American plate
Yeah,
side.
so we're at a subduction zone where one plate is sliding
And the
under
Nazca
the
plate's
other.
the ocean plate that goes underneath.
Yeah.
That's what forms the trench.
Oh, the fish are stream avatars is the word for them. I do have the proper credits in my Twitch description. I don't remember off the top of my head who made them, but it's Stream Elements is the program. No,
I'm so sorry that in
Stream
my excitement,
Avatars
I
is the
forgot to
program,
introduce
and
the
then
students.
I got the fish from the
So
workshop.
we have a number of
April,
grad students
hello.
in here who
Hi.
have been doing incredible
Welcome.
work throughout this entire cruise.
I am also multi-streaming, so I apologize.
We
I'm
have,
talking to some
Miguel,
people on
who's
YouTube,
a
some people on Twitch.
microbiologist.
Thank you for your patience.
And we have Katie and Amber, who are astrobiologists. Well, they're all astrobiologists. Katie works on space biology.
Yes,
She's interested
yes.
in producing food for astronauts on Mars.
So yeah, this is they have such cool people on this. These dives are always super cool, but this one is something
Put on
special. Look
Katie's
at
mic.
the,
I want
look
to hear
at
all
the-
about what she does.
Unmute.
There we go. Now I can.
Hello, I am the Katie that has been mentioned. Yes, I do study, unrelated to this, I study sustainable space-based agriculture, but
That's so
I also have
heckin'
interests in life on other planets, and that's kind of how I find myself here.
Hi, Katie.
Thank you. Hi, Katie.
Amber.
Richard Vilo, look, Ma, I'm on TV. Viro, hello.
Have another set of headphones for Amber?
Look at that skate. Look at that boy.
Can we get Amber a headset? Do we have extras?
So we are 2,800 meters down. So that's like, I think someone said 1.7 miles. We are really deep. What's the difference between a ray and a skate? You know what, Katie? You know what, Kate? You got me there. I don't actually know. I need to play more Fathom first and refine my skills.
The deep sea flappy thing is a skate.
The deep sea flappy. There we go. That's how I'll describe it from now on. The sea flap has a skate.
Amber, why don't you introduce yourself?
Hi, my name is Amber Wu. I'm an astrobiology student and I study the formation of cell membrane before, I guess, it becomes a lipid membrane.
Ooh. Very cool.
Also, I also
Miguel
have
is here with us again.
Quail.
Yeah,
Quail cam.
I've already been talking a decent amount these past few days, but just in case, I'm Miguel Arribas.
I will close that though because we're looking at
a student
the
from the
3D
Spanish
model
Center
right
for
now.
Biology. We got some more methane clams, which was nice.
Was that the one we started with? Okay. Yeah. No, sorry, Miguel.
Oh, don't worry. And I'm also technically an astrobiologist.
Deepwater quail,
My
yes.
PhD is more focused on biochemistry, although I'm mostly doing microbiology in here.
A little stinks. They actually don't smell yet. They smell kind of nice. I imagine
Why are
as
the
babies, there's an
organisms
incentive
that
not for
seeps
them
and vents
not to
white,
smell.
like the crabs. Nick, do you know? I'm not sure about that.
Okay, we're gonna. Kate, Shining Deer, looked up the difference between skates and rays. Okay, according to the Florida Museum of Natural History, it is Florida. But they would know about ocean critters. Most rays are kite-shaped with a whip-like tail possessing one or two stinging spines, while skates have fleshier tails and lack spines. Rays protect themselves with these stinging spines or barbs, while skates rely on thorny protections on their back and tails for oh, so rays are the ones where if you step on them, it's a bad time. Nature's chicken nuggets should be hard to detect. Yes, yeah.
Smooth sailing a few meters above the surface.
Yeah, we're really.
Scotty's talking about the quail.
Flying,
They're
really.
very
Flying over the
zoomy,
seafloor.
and also at the absolute bottom of the food chain. That's
I've
not true.
never
They're
found
actually
a whale
insectivores,
carcass.
but
I'm sure some of the folks at Schmidt have found a whale carcass at some point in their lives.
yes, it's out. Scotty, did you step on one once? Yes, it's ouch. My foot still has a numb spot. On one hand, that's
I have a
kind of
request from
cool,
my
but on the
class
other hand,
students,
oh no, I'm
to find
sorry.
a whale carcass. So
That must have been a bad day.
How is Sebastian powered?
Umbilical. I know the answer to this pop quiz. Umbilical.
By Whimsy.
By Sebastian is powered by whimsy. I failed the quest. The answer was whimsy.
It's connected to the ship. Runs on electric.
I see that I've been specifically asked a question from the Facebook chat. An idea
Skate
for
is a piece of
launching
foot
through
gear
and
for
how could
moving
you find
across
out
fast
if
ice.
it's
Array is a beam of
I
electric
actually got
radiation.
into
Hope
it.
this
You could
helps.
Google this yourself. There is an international competition held by the Institute of Competition Sciences called Plant the Moon Mars.
Plant the Moon
Competed in
Mars.
that
What?
For 3 years, and that is a global effort of trying different crops and different methods of growing in simulated Martian as well as simulated lunar regolith.
We're gonna do this, chat. We're gonna do this. We're gonna plant the moon.
The simulated regolith is also commercially available through the University of Central Florida Exolith Lab.
Chat,
You
new
could
stream
purchase
activity.
your own regolith and try it home even.
Loren, look at those sponges.
Oh wow, pretty sponges. Can we zoom in on these real quick?
But my recommendation for you is to look into literature and just give it a try. It's a lot easier than it looks.
We may want to consider
We're gonna
collecting
do that.
them
We're
because I haven't
100%
seen them
gonna do
anywhere
that.
else. It's up to you.
Yeah, those definitely look
Once I
different
get the
from
actual food
those
garden
sponges you've
in.
seen. I'm
My
happy
garden
to
cleaned up. I
stop
wanna
and
get
take a
yard birds.
sample.
Yes.
Yeah.
Get the info on the simulated regolith. Yes. Also, look
Take
at
a couple
that.
sponges for Nick.
Oh, there's what's that kind of purple thing? There's stuff happening here.
I was also hoping to get a couple of these crinoids at some point, so.
Oh, and there's the crinoids
here
are
was as good a time as any.
the branched things there.
Ian, you're back. Did you see the seat?
You're welcome.
And they do look like
Yeah,
little
they're
palm trees.
hexagonal
It's true.
sponges.
And then they swim, and it's like, what? You can do that?
Hello to Andy Portalatin from Texas, an ex-student of mine.
No, hi.
Bought a California Reaper plant yesterday. Gonna pot it and hope for the best. Let's go.
Yeah, I'm thinking of grabbing a couple
Need
of
to
those
finish the
crinoids
Florida
since we're-
room. Wait,
since
Rick,
we're
what's the
stopping.
Florida room? Rick, elaborate. I'm afraid.
Another great living fossil, crinoids. They appear in the fossil record 300 million years before the dinosaurs.
300 million years before dinosaurs. Crinoids are just like, all right,
Oh,
I'm
a whole
good.
bunch of people giving shout outs to Nick. That's great. Aw, you missed it. We had a big moment in here a few minutes ago, Ian, but it's okay. We're gonna go back. We found a methane seep. So we're just kind of scouting out this area to see if there's evidence of more before we start sampling. Ask Nick. It's for him.
If you can pick up 2 of those tubes.
Sure.
Entire sponges?
Yes.
It's mostly
but we don't need to.
gauged
to
in
take
room
the whole
currently
thing,
full of
just
junk
2
in the
of
back.
those, 2 of the
Oh,
tubes.
got it.
2 of the tubes.
Crinoids
Okay.
have reached their peak
What
and are
would
resting
be
on
the
their
best way
laurels.
to grab
Yes,
those?
yes.
Suction?
Crinoids are like, all right, I'm good.
It's possible
Manipulator?
to manipulate
Okay.
to get them both, maybe.
Okay.
Oh, I'm
Okay.
dropping a
Yep.
lot of frames. I'm gonna.
I don't want to take the
Oh,
whole
We
lot.
sorry,
don't have,
guys.
we haven't put anything in any of the bio boxes yet, so.
I'm gonna end stream briefly and then
Really,
restart
whatever's
it. That
easiest to
usually
put them in, I
fixes the
guess.
dropped frames. I'm so sorry. I will be back in one moment. I
Probably one
lied.
of
We're
the
gonna wait
smaller
for the
ones.
sample to
They're
happen first.
dedicated.
I don't want to
Maybe
cut it
you
off in
can
the middle of the
consider
sample.
coral cleaver.
A coral quiver? We can do that.
If we get any bivalves, they'll be very heavy.
Yeah, okay. Coral quiver it is.
If
It's not noticeable? Okay, alright. I might leave it alone then. It says it's at 11 I'm gonna stop.
Can I
They're gonna
actually
sample
get,
it. They're gonna take it
an
home.
extra sponge for me. I'll sequence the microbiome. Never seen this before ever. Take 2.
We don't want to take the whole animal. That's
No, I
why
don't
we're
wanna
just
take the
taking
whole
a,
3,
a
yeah.
partial sample.
Yeah.
Shoot, I
Hey
did
Danielle,
not fix
how are
it.
you?
It's actually
Good to hear
worse.
from you.
Rip.
Hi Griselle, how are you?
Picks up the sea pig.
Good question for you, Nick. How
Yeah,
do you
they're gonna-
decide what samples to take?
This is for my students, so I choose not to answer any questions from my students. Hi Griselle.
Maybe I won't kill a
So
kill
the
the
extract
stream.
mineral sponges is
Have to
not
act
just we're taking
decisively.
a sample for the sponge itself. It forms a habitat, and sometimes the association with other crustaceans is very species specific. You may find shrimps that just completely associate with the sponges and are not found anywhere else.
Wow.
So it's not just the sponge,
Giselle,
it's
for
what
your
might live in
question,
the sponge.
sampling, well, okay. So we have to go back. Every day is planned the day before by the principal investigators. I mean, they take ideas and then they form a plan. And we follow pretty much the plan, let's say, to the dot, unless something extraordinary happens.
Ah, yes, my weekly ocean speculating stream. Wonderful way to conclude today. Welcome, enjoy.
What we do is
We're getting
we
sponges.
have some idea of what we want to sample and where we want to sample, and we just follow those transects that they were laid out from the night before when the ship maps the general area. Okay? Now remember, this is a discovery expedition. We don't know exactly what we're going to be finding, so a lot of the actions are determined on the spot. Okay?
Yeah, I don't think this area's ever been dived. I think this is
Species
like
ID, as you know, for your projects,
Never seen
for the stuff
before.
we do in the lab is difficult. Unless you have an expert, for example, if you got an expert on vents, this person will help us more. Most of the work to take it down to species is happening in the lab. And other studies, this isn't the first ship that has been found in Chile. Somebody just
this is
sent us a
happening live.
paper on the first report of a ship in Chile that was 20 years ago. However, for our group, this is a very exciting moment. This is a very rare habitat, and if you have been watching the video, this is a relatively it literally a needle in a haystack. Okay, this is complete by chance. You should have been here when the lead group discovered the habitat.
It really was something. Even with all of the bathymetry data and everything else.
Yeah, the ocean's
we were
big.
Really taking
They
the
were
chance.
very lucky to find the
Finding out a
methane
course
sea.
and just hoping that we tripped over one. So that's beautiful. That's beautiful stuff.
That is a beautiful sample.
For the sampling itself, we of course we try to be extremely mindful of what we're collecting, and
let me do another shout
we do
out
understand
for
that some of these species
Wows,
are very
because
rare.
I was really hyped up.
So
Guys, if you don't
keep
already
that in
follow,
mind, okay? That we don't make, we don't collect.
I always say Mola.
There's a reason
Mola
for every sampling.
I get grab one more for me.
is more. Please follow.
One for me. Yeah, same.
Mola does art.
Okay. Yeah. Just one for me to do the microbiome. And we'll get a shot of it as well. Make sure you get a picture. Glamour shot.
Mola was doing Elden Ring. It also does art.
It's a beaut. Thank you much.
Wait, are other folks getting a stream? Oh. Mola.
Yes. Let you.
Yeah, I killed my stream in an attempt to salvage my dropped frames, but they're just slowly getting worse, so I don't know what to do. I might unplug my hands.
We have hours and hours. Ooh! Isopod!
Oh! Isopod! Oh!
Yep, we'll just keep jumping around. Okay, once... Oh. My finger was caught in my headphones. Once we get this...
Yeah, it's a fast one.
Last sponge sample, we would like to get some of these crinoids as well. And put them in a different box. I think a bio box.
Just needs to
would
pull
be
the
more
quill
appropriate.
feathers out of her CPU fans, probably. I like isopods, they're cute.
It's tough to sample these and to be delicate when you're using a robot arm that's literally thousands of meters away.
Yeah.
Nah,
Yeah, they're doing a
alright.
really good job.
We'll leave the rest
And you
to
Pilbug
regrow.
holds gently. Precious Pilbug.
Yeah, you can put it in the coral
We should
quiver
use
if it'll fit.
Pilbug in our moon farming simulation.
Yeah, one of the smaller ones. Just thinking the clams are probably gonna need to go in the bio box. That's good. Yeah.
So cool.
How do sponges regrow? They don't really have tissue differentiation, so you break a piece off, it can grow back.
It can grow back. That said,
They have different cell types.
usually
but
do
you're
grow
correct. Yes,
really
they don't have
slow.
tissue differentiation, but they have several different types of cells. Plus, they have structure. They have spicules inside their tissue, which makes them keep the form that we
almost
see.
guarantee it's twitch related.
Gives them the structure.
The not restarting issue.
Similar to, let's say, the iron, and you lay down the iron and then the cement, something similar to that.
Yeah. Rebar.
Yeah, rebar. Yes.
Yeah. Reinforcement, yeah. So they do have sponges do have different kinds of cells. Oh, we're just trying to mash this guy in. Let's get in there. Get in there. Look at the shrimp. I think that was a shrimp. That actually might not have been a shrimp.
Sponges do regenerate. We know that there have been several studies already. You can cut a patch and you can follow how they replace the lost tissue. So indeed, since they don't have specialization in the tissue, they can regrow the lost area. Okay.
Just I feel so bad for this sponge. Just trying to pat it in. Water has discovered my stream deck plays games. Uh-oh. Her stream deck
So
now.
instead of the audience asking
This is a
us,
joke.
I'm going to ask Andy. So since he's listing two of the different cell types that you can find in the sponges, guanocytes and pinacocytes. Andy, what other cells are we expected to find in sponges?
I also
You don't have to
hope we
answer
don't
to
get-
me. You already got my master's, but the master's for me. But just for the audience.
Oh, is he roasting his students in chat? Oh my gosh! I think his students are in chat. Pop quiz.
For his master's, Andy used machine learning to identify sponges from pictures from the web.
Oh, wow.
So that's he was. We're trying to submit it in a journal right now.
Mentioning machine learning, Fathomverse is a game you can play to help them build a
So
database
next
to
we'd like
identify
to get some glamour shots
ocean
of the
critters.
crinoids and then sample them, please.
It's available on Android and iPhone. I have to see if my professor is doing something like this. They would totally be in chat,
have
right?
chimed in and given us a potential flight pattern spot to look at. So I'll show you where once we get those crinoids. Rad. Love
No, man, my rig is pissed. Wait, I'm not supposed to say that. My rig is very upset.
Stocky boys. Yep, stocky boys. What if a flower had a skeleton?
Some sponge divers try not to cut all of the tissue on sponges to preserve the fishery. Makes sense, makes sense.
Okay, first of all, when I get let's get mix them.
It's like
Yeah, I
it's
gotta
like
see them
a windmill.
I gotta see them. This is a different species. You can see the.
Okay.
This is
Okay.
a little further away.
All right, can we zoom out a little bit for Nick? Nick needs to identify. He says there's two different species here, actually.
Yeah, they're
He'd
so
like to get
pretty.
both.
I really like them.
Mm.
I think on the side, just looking at this, but that may be a possible title.
Oh, okay.
Some crinoids can swim. I wonder if these are able to. What if a flower had a skeleton? Lab staff, trying to figure out how to rig this model.
Okay, so each
That's a
one
joke.
of
It's
each
a
of
V,
those-
right? It comes this way.
Should they go in a bio box? I don't know if they'll fit in a quiver.
It reminds me how Melissa would always say,
Bio
"Can I
box?
get a
Okay.
quiver size sample?"
Okay, so we need. Hi, babe. My husband just jumped in. How are you? All right, I will catch you up in just a moment. We need this crinoid and this one. There are two different species, and they can go in a bio box. They can go together, right?
Yes.
Okay, they can both go in a bio box. So, if you're just tuning in now, we found a methane seep moments ago. It looks like it is potentially on the decline, but we did see little teeny bubbles of methane coming out from the sediment. And we found a whole bunch of clams and tube worms. And so we marked those on our map, and we will come back to sample them. But first, we're going to move a little bit further ahead and see if we can find some more activity.
I hate to put it this way, but the lighting and composition of this pic,
We're just repositioning
the
ourselves
crinoids
so we
is
can sample these
aesthetic.
crinoids
No, it really
for
is.
Nick.
ROV pilots are like also artists, I swear.
We're not in a submarine. This is a remotely operated vehicle.
Yes.
So it's a robot that's connected to the ship, and we are controlling it from on the ship. So there's no people inside of Sebastian.
Yeah. Sebastian is an ROV, remotely operated vehicle.
That's wonderful. So exciting.
That must have been a Facebook comment because I'm not seeing it in their YouTube chat. They are also multi-streaming. They are streaming to several places.
I love crinoids. One of my favorite.
I feel slightly
fossils.
listled. Thank you.
Every time I see
No.
a fossil crinoid, I'm like, oh, look at that. It's so beautiful. I want it. Just getting ourselves into place here so
the
we
babies
can
are chorping.
sample these crinoids
Chorping.
so that Nick can study them.
You're okay, little ones. You're alright. Did one of you poop directly into the food? You did, didn't you? I lied. You are stinky.
Would it be possible to take an extra of each one so I can do some microbiome sequencing on it? I don't know how many of each species are here, and I don't want to over sample.
When do we get pet crinoid? Right now, we're taking the sample. It's happening. So because there are multiple science teams, that do like different things, someone requested a sample of each kind of crinoid, and then she's asking if she can also get a separate sample to do her thing as well. They're separated from big birds? Are they being mommed? No, they are separated, unfortunately. As much as she's laid a bunch of eggs, none of the adult quail are particularly
Yes, bio box,
parental.
please. That one, Nick, can you point these out again for us?
Let's get the tall one.
The tall one?
I have a message in the YouTube chat from my partner. Hi, Ty.
Aww.
Thank you for stopping in.
Let's go to grab it from the as close to the base as possible. It should be easy to break.
Yeah, right. Close to the root, the test. What?
Base.
Not test.
Hey, don't forget
Base.
to.
Just
Oh
base?
yeah, pre-download Genshin. Yes.
Not test. What was the word I was thinking of? Hold fast, I guess. Nice. Nicely done. Ooh.
looks like we might get that,
Pretty. Oh yeah, I do see
its
the
little
difference
friend as
between
well.
them. No, wherever we think it'll fit. It's kind of a long boy, so. A crinoid is a type of echinoderm, so they're related to sea stars.
Cry about it, Noid. Real crinoid hours.
But they are pretty much living fossils. They're very, very ancient organisms. So we have fossils of crinoids that are, I think, appeared 300 million years before the dinosaurs. So,
oh, blueberry and calibrate. Sit up straight.
Crinoids have been around for a very, very, very long time.
And stretch. If you're sitting on your
A
legs,
skinny
stretch
sea
them out.
cucumber with a brittle star attached at the top. I don't know if that's accurate, but I like it. I don't know if that's anatomically correct, but it sounds good to me.
Visually,
How
I
big
can
are
see
the
it.
crinoids? They can get pretty big. I've seen fossil ones that are huge. I don't know how big modern ones get, but
If you're sitting on
this
your
one's
legs,
pretty
consider
long.
standing up and doing some little duck steppies.
We may have a bonus organism of some kind on the crinoid, but I can't see it from here.
Once you are done, flex
Amphipod?
Yes.
Yeah.
and dab. Whoa. I know where. I'm gonna hide myself while I try to adjust my hands a little.
So yeah,
you guys watch the.
My husband is now on the stream. If you could just tell all of our friends and loved ones that I found a methane seep, that'd be cool. I do it myself, but I'm a little occupied at the moment.
Kind of on an expedition. I'm gonna try to make
I think the other one he wanted is one of these guys. Can we zoom in and get a little picture first? I keep forgetting to ask.
There should be a way for me to
Yes.
adjust.
That one. There.
Yes,
right?
here we are.
Okay, yeah, this guy.
The area for my hands.
Yeah, I can see what you mean now. That is different. The branching is different. Yeah, beautiful.
Okay, so I've got that wave.
Oh, thanks, Ian.
I don't think I can
Thanks for
fix
sending
how if
back
I
the
can
message.
catch ducks, but I should be able to fix
I had no
the
way to
cookie
notify
disintegrating.
everyone, so I just sent a hurried
so
text
far from my face.
to Keith to get on the stream. That's beautiful. Thank you. Yeah, let's try to get as much of it as we can.
Guillermo just mentioned to me that there has been reported only one crinoid species in Chile, and we have already doubled or perhaps tripled that record.
Oh wow, that's awesome. So we've found a few new living fossil species today as well as yesterday.
That's so cool. That's so heckin' new species. Let's go.
That's outstanding. I love this. This is beautiful.
I wonder if the currents weren't as strong. Wait, hold on. Oh, like, that's why things got bigger? Maybe.
Same bio box, yeah? Yes.
Yeah, I think they're going the same way.
Okay, so that should be more adjusted. What in the world is that for?
So pretty. Very pretty. Now, if you were here earlier, you
Oh, that's
might
my
have seen
hitbox. I
fields
have a
and fields of
hitbox.
these. Almost like a field of flowers, all pointing in the same direction to capture food from the current. Oh, okay. Give the babies a kiss for me, hon. Love you. My husband is at home with our 5-year-old and 7-month-old right now.
Okay.
Oh.
So apologies, let me readjust myself a little more.
Do we want that as well?
Oh shoot.
That's what
Okay, there. Sorry.
Okay. Can we also take? I'm sorry.
Yeah, that was my fault. That was my fault.
One
That was
more
my
of
fault.
these crinoids for myself for the microbiome. Doesn't matter.
According to
I think
Wikipedia, those
these are all
deep
the
sea
same
species that
right,
retain a
Nick?
stock
Those are all the same.
1 meter,
so any
3
one of
feet in
those,
length,
is
usually
great.
much
and
smaller.
Then Nick wants this sponge for
Fossil
the museum.
species, 20 meters, 66 feet.
Yeah. It's a question like, do they have an exoskeleton?
Okay, so let me see if this works now. If this works a little better. Can you guys? Now you guys can see the cookie. Before I shove it in my face. Let's go. Rick Muppet says crinoids are made of crinoidium. This checks out. It checks out. Cookie Monster Cynthia's. There we go.
Oh. Yeah, same one's fine. Thank you.
So they are. Again, this is another place where depth comes into play. They probably actually are dealing with some lag right now. And she's have to say, oh, no. Oh, okay. Wait. Is it on a cooldown? It might be on a cooldown.
Oh.
Ah, it's on a cooldown. Alright, that's fine.
I think we're getting a free hitchhiker here.
Oh, pardon me. Sorry.
Amphipod. There we go. We get a little amphipod for free. And the sponge can go in the same bio box.
Yes.
Thank you.
Yeah, like with the sponges, sometimes seeing what is associated with the sample is also very important. Sorry, I moved stuff around and now I'm like, how was I sitting before? How was I think I was sitting like this. Okay, sorry, it probably froze again because I'm... Oh, wait, no, I'm bad!
Bottom.
There we go, okay.
Do we have a pH sensor on? No. No. So, the average pH of the ocean is slightly alkaline.
Oh!
So yeah, the pH of the ocean is slightly alkaline on average, around like high 7s or low 8s.
Yeah, that's a big...
And yeah, ocean acidification is certainly a threat to ocean life in general.
Gosh darn, this wasn't my fault. Aside from me not having my rig upgraded. There we go.
I don't know. I know that it leads to things like coral bleaching. I'm not sure how it affects sponges and echinoderms. But...
Well, I think ocean acidification affects the availability of calcium, right? Ignore me. I don't know.
Ah.
That is a nice sample. Gosh.
Yeah, some of the fans in the live streams are extremely knowledgeable.
Yeah, they're probably talking about their
Certainly more
YouTube.
so than I am as a microbiologist. I only know how to ID these organisms in a very general way, which is why we need folks like Nick and Guillermo to help us out.
Sorry, the babies.
Yeah, we got some nice specimens here.
The
Okay.
babies are zooming.
Pilots, you know where you're going next?
Lambda, you okay?
Oh. We have a request to zoom in on this rock over here quickly.
There was a possible chiton.
Maybe a chitin over there.
for Lisa in the audience. I'm not confirming. I'm not expecting any, Lisa.
Oh my gosh, I'm so sorry. Ow, 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 ow.
So we want to kind of go to the left of this ridge towards that next waypoint.
Okay, so stay to the left.
Yes, please.
Head up down. Okay.
Yeah, that's what the geologists are saying.
Okay.
They've eaten all the wet mash. I should probably get them some more.
Can we go over this?
Yeah, hungry babies. They have a lot of growing
I
to
got
do.
called a young lady. Thank you, that's very sweet.
Dr. Lauren Saylor is the lead person, and then there's a group of 2 or 3 people around that have.
Yeah, hungry babies is a very good thing. It's when they stop eating that
Yes,
you have
we
to be
have
worried.
Quite a
Like,
few
really worried.
amazing students, graduate students on board this expedition who have been absolutely invaluable to this work.
Hold on. One of them looks like he's having a hard time with something.
So we have Amber, Katie, and Miguel in here right now.
Hey, are you
Yes.
okay? Oh my gosh, give me a heart attack. I thought
This
you
was
were
supposed to be
choking.
their
Jeez.
dive watch, but I
Oh,
sort of had to
give me this.
take over for a moment.
Refill it later.
It's okay.
Your
Drew
stinky
said we can
babies.
stay through his.
Aw.
Small idiot
Drew
bird
is,
child.
Drew is a mensch. He's a good guy.
Did you see Drew?
Yeah.
I'm right here.
You're a good guy, Drew. We love you.
Oh, I love you guys too.
Aw. All right, where are we looking? To the left? Here, let me give you the pointer. Oh, that's right. Getting your left and right mixed up. No worries, we're all pretty excited right now. Zoom out a little bit. I don't see it. Maybe it was that.
These bird children.
No? Something
These
different?
stinky little bird children.
Oh. Oh. Oh, is it that down there? Maybe? Let me zoom in the bottom left here. At the base of the crinoid.
Oh my gosh, they
here.
found
Maybe that was
the wet mash dried out and there's wet mash flakes. No, let me get those out.
That's a
I might
sponge.
have to just go get them some more wet
I think
mash.
they're both sponges. Okay. All right. Let's keep going. We're gonna go up to the left of this ridge, because this looks like a potential place to find more seeps. Possibly they'll be more active. But in any case, we've marked the location of the first ones that we found, so we can always come back to those.
Sorry. How's that? Is that good, Tyler?
To the Twitch chat, we are very excited to be included in the mission control room. I personally have spent the last few days in the wet lab filtering up near 200 liters of seawater, and because our vehicle is down for 24 hours today, I have the time to come have fun.
Yeah, this has been, because this dive is so long and we haven't taken any
You're the whole
samples yet today,
frustration.
it's been a very relaxed day for us. We've just been flying across the bottom of the ocean, keeping our eyes peeled, and our patience has paid off.
Like I'm not
Very
actually
exciting
upset,
to
but
have
also
everybody here
you
to
little stinkers.
Tomorrow's going to be a very exciting day to see all the sample surface.
Yeah.
Okay, understood.
Thank you.
Wow.
Updated waypoint.
ROV watching as a hobby sounds like
You tiny, wretched, beloved beings literally made of these stinky birds. Not even
put them on
stinky
in the
yet.
background while I'm doing stuff. That's fantastic. It's so great to know that there's fans of SOI that are so dedicated. The meteor-like rock collected yesterday.
Same with
I
my
wasn't
cat.
on
Yeah,
dive watch yesterday. Does anybody know what rock they're referring to?
What about your sticky rock from yesterday?
Sticky rock from
The one
yesterday?
that was hard to get? Yeah. You have a question in the
You're
chat?
asking about it in the chat.
We're asking about Miguel and his rocks.
Tell us about your rocks.
So answer, it was not a meteor. We looked into it.
Oh, they must have collected something that might have been a meteor. You just said it's not a meteor.
So it broke upon collection. It had some metallic parts inside, but most certainly not a meteor. I have kept it safely, so.
Very cool. Someone was asking what keeps the water in the Niskin bottles and the bio boxes from popping out like soda at sea level pressure. We do rise pretty slowly, so I think that it has a chance to equilibrate.
Those bio boxes are not 100% sealed, so there's always.
Yeah.
They're not air.
That was me. There goes Rick.
Exactly. I mean, not the bio boxes. The Niskin bottles, I think, are pretty watertight, but we do rise up pretty slowly, so they probably have a chance to equilibrate to the pressure. Yeah. We're aiming to
Answer my question in less than a minute.
This side of the ridge, so to have this like on our right.
Yeah, if we can. Yeah, if you can aim to be in that little divot there. I like thinking about how dark it would be down here if we just turned off all the
Your
lights.
mom beat Storm Terror? That's pretty
This
cool.
whole world in darkness.
Spooky.
It's very spooky.
Also, it's gratifying to hear that they also would find it spooky in the complete dark at the bottom of the ocean.
Have any
Confirmed
clay
spooky
samples been taken?
by scientists.
Yeah, I would say that our sediments have had some clays in them, for sure. The geologist could speak to that better than I can as to what the sediment cores have looked like so far.
I can't say with certainty, Ty, but I think some of what I'll be bringing back to our lab will have some. Once again, can't say with certainty. I'm not a geologist.
That's the buzz phrase of this entire cruise. I'm not a geologist, but
I guess the geologist went to sleep.
Drew says hi, Ty.
We're just gonna make our way alongside of this ridge here. The backscatter data for it looked really promising. So.
People in the chat are questioning how cool the uniforms are, the ones that we have, and they are comparing us with the cartoon show called Sea Lab. And I suggest to the PIs that we need to have a nice uniform, encompassing the mission.
I agree. I would like to have a speed suit like on the Venture Brothers. No, it's not a jumpsuit. It's a speed suit.
This crew.
Oh, that is a shame. Venture Brothers is a great show. It's a very, very funny show. Hi, Dad.
Making our way as if proceeding downtown, walking with haste.
My dad is in the chat.
Rock
Hi,
faces
Dad.
pass. So we're homebound.
Everybody says hi, Dad.
Hi, Lauren's dad.
Hey, Lauren's dad.
Oh, the forbidden calamari. I did see that get captured. That looked pretty gross. I wasn't there when they took it out of the ROV, but I imagine it was pretty stinky.
It actually didn't smell very bad, and it still had its beak completely intact.
Oh,
wow.
So we were looking at that for a little bit.
That's cool. It didn't look like it was that complete, so that's awesome that you got the that you got the beak and everything.
Its tentacles were a different story, but
yeah. Well, I saw those were like broken off or chewed off or something like that.
What they're talking about is a squid corpse they found where it looked like the tentacles had been severed. Not chewed off, but like cut. And the only thing that was left was the like head portion, and even that was kind of chewed up. And there weren't a lot of scavengers, so it happened relatively recently. So it was this little mystery that I guess apparently they're calling the forbidden calamari. I'd be curious, like if it was like bait that fell off a hook or something?
We did not slurp the forbidden calamari. We pinched it. We grabbed it with the grabbers. That looks like more biofilm. Can we zoom in
That
here?
does
and
look
look
like
at-
more
What are
biofilm.
those little? What are those little guys? There's some clams here. but it
More
looks like
evidence of
shells and-
methane.
Ooh. Fish?
Fish?
Those are fun.
Long dead bivalves.
Yeah, those are
Yeah.
definitely toast. But there's some biofilm looking material here. If we could get a shot of that.
Yeah,
Log
yeah,
it.
yeah. Fart smelling fish, no!
Sticking out of here. I don't know.
But
what
also,
those might
you
be.
are technically correct.
Maybe crinoids. Yeah, drop a marker here. Maybe we can come back to it. Because this is a pretty extensive mat.
Yeah, that's a lot.
Well, that's up to Aparna if she wants to take a sample here or somewhere else. Okay, Aparna would like to take a core here.
Yeah, I guess might as well. Instead of depending on doubling back.
Yes.
So they're going to take a
Yeah, in the
A soil
highest
core.
concentration of the white here. Here, yeah. Oh, sorry, I pushed too hard. And we're gonna have to be gentle, so as to try not to disturb that top layer there. Yes, please. Start with 1. 1 and 2 are reserved for Aparna because she needs to take her cores off first. I did, yeah.
Sorry, I'm also peeking down at the children. One of them, I can't really tell which one it is. But I think it might be... It actually might be Lambda is, kind of grooming themselves. Which is really cute. I'm not distracted.
Okay. Can we just go very, very slowly on this? Thank you. I'm gonna try not to disturb that top layer if we can.
For Andreas, that is commenting on the chat. Bacteria are the beginning and the end, not just the end.
Yeah, that's a very good way of putting it. And that applies to lots of things, not just methane seeps, for sure.
As they get into position. So, I've been trying to wake up a couple times. I want to just talk about, like, the quail and the trials of the quail. They sleep on a heating pad. I don't use a heat lamp because of the associated fire risk. And the heating pad does just fine. I think there's kind of a push to move away from heat lamps and towards other methods of keeping chicks warm. But one of the benefits and the detriments of the heating pad is the quail can move closer to it to be warmer or further away if they start to overheat. But that means they can stumble off the heating pad. Which B did the first night. And I think being cold was almost a very big problem. And last night, one of the others, I didn't even see who it was. Yes, there's also the risk of overheating with heat lamps. What I think the popular thing that's kind of being used as a replacement now is there are squares that stand on legs and they heat, and you can kind of angle them, and then the chicks are encouraged to go under them. But for me, for my setup, heating pad was easiest. Last night, someone else stumbled off the heating pad and then tucked their little head, between the napkins that I have covering it and the heating pad cover, and that probably would have been fine. But there are places in the heating pad where they can get where they can get stuck, and it would be a problem.
I'm sorry, that just made me laugh. It's got nothing to do with microbiology or marine biology, it just made me laugh.
And it's one of those things where, like, you think you've got everything perfectly safe, and then the quail will just figure out a way for it not to be perfectly... They're absolutely running laps right now, by the way. Let me turn it on. Little stinkers. You can almost see, even the babies have crest feathers. You can almost see the crest feathers on that little one.
Yes. Aparna, can you show us exactly where you would like this? This is your show.
Oh gosh, that one just has the absolute zoomies.
Yeah. Yeah. That's
Yeah,
a
everyone is
spot
so
right there.
strong right now. Yeah, twice lab staff has caught them off the heating pad. Bless lab staff.
Oh, a little over to the right.
There we go, just that really thick.
It is definitely
that
a bit like a
really
space
thick
operation.
portion. Sounds like a good design. Yeah, radiant
You're
heaters. Yes.
Using a remote.
Also heard of fake rocks that are heated for lizards. Yeah. Being used for birds to warm on. Oh, it makes sense. I mean, it's designed to be safe for reptiles, but it could very easily be repurposed for birds. Or not even repurposed, just used for birds.
Okay, yeah, we're trying very hard not to disturb this top level layer here.
Yeah.
There's another clam there over on the left too. Suction it? I don't know.
That shrimp was almost part of the core sample. Not a whole lot of difference, if you ask me. I mean, birds
If it goes
are
into the
descended from dinosaurs.
suction,
After
it'll
yeah,
go
yeah,
into
yeah.
one of the jars,
As long
but
as you
there's
keep
a
the
mesh
cords
at the
buried,
bottom of those,
don't
so
let
I
the
don't-
poop build up. Should be safe. Yeah. For the heating pad, I have it partially. I have part of it. It's in an L shape.
Right.
They sit on the L shape, and then it goes up the wall of
putting it
the
into one of the suction
container, and it's
sample jars
velcroed
is
into
the
place.
best way to do it. I think a core would be
Bottomless shrimp
better,
sample.
probably.
And the one I got specifically is waterproof, so I can like wipe it down if I need to.
Right. Maybe if you just take
Red lobster will never
instead of pushing
recover.
all the way down, maybe just
Hey
take
Velcro,
the
it's
first
it is
little
super
bit.
useful. I have some like fairly heavy duty Velcro I use all over the place.
Yeah, she really just wants the top layer, so.
And then I also I bought a bunch of like Velcro strips for
think right
cable
about there,
management, and they
please.
get used everywhere. They're so useful. Alright, so they're gonna try to take a core sample without disturbing the top layer. It's kind of a delicate thing. I thought that whitish thing was dead, but I think that's a living worm? Oh, there we go. That's
beautiful.
Yeah.
Awesome.
For the size of the mess, that matters. Of course, it matters. That really depends what the individual researcher wants. So a mesh for one person may not be suitable for a mesh of the other person. But you are correct with pointing that out.
We just, I think, very, very carefully and then gently lift.
Oh, it's not gonna stay. Oh, it's too soft.
Oh. I I think this is the first time we've actually had a failed core.
Oh, it was a problem on one of the
Material here
previous
is just
expeditions.
so loose.
Yeah.
Oh, I'm wondering if there's a better way to do this.
Cause they really want to keep
gonna be the way.
Yeah.
Oh, maybe it's still in there.
Oh. Is it?
I don't actually know. I don't think it.
Oh. Oh.
It didn't.
It stayed.
It did
Let's
settle.
go. Okay.
Okay. I thought we lost all of it. Wow. Okay. Sorry. I won't jinx it. I'm not gonna say a
Yeah.
word.
Like, fingers crossed. Fingers crossed. Ah. Oh, maybe they did. Is that just sediment in there? It's so hard to tell. It doesn't look like it's not moving like just sediment.
Is that just fluff?
Oh, maybe it is.
Oh yeah.
Oh, it is. It's all just.
Oh yeah. That's all like suspended. Yeah.
You're okay, little ones.
What about the scoop?
Maybe the scoop.
Yeah, that
but
does
then
stink.
That's not gonna keep it intact.
Correct.
Yeah, they were really banking on getting these samples. These samples are really important. They could try sampling in a different place where it might be a little thicker. They've had to do that in the past where they just really want to sample from a certain area, so they try a couple different places
Could
for
sediment
cores.
scoop it, but I don't know if the top layer will stay.
Yeah.
The scoop, yeah, the scoop
Stay together.
Look at all the. Okay, I'm gonna turn off the quail cam real quick. Look at all the fibers in the water now. Oh man, it's like somebody's grooming a husky. That is a whole bunch of bacterial matting.
Try down there.
Yeah,
Maybe
that
it'll be
is
a
a
little bit more fun.
lot.
We can try it if you want to.
Yeah, I think they're gonna try a different spot.
Right about there. Okay.
Oh, overlapping?
Yeah.
I would have thought they would move away.
Yes.
What is the core sample? What the core sample system needs is self-closing. Right. The problem, I think, is that it's really. There's definitely value in keeping things as simple as possible. But then you have moments like this.
Oh.
Oh. Uh-huh.
Alright. Just have to be slow and patient, I think.
Yeah.
We'll see if it pays off.
So it looks like they're sampling in the same area, but they're going to let it sit for a minute to hopefully settle out in the
For the audience, isn't lubriculus?
Do you watch Girl with the Dogs? Huskies are. They are! I've watched a couple of her videos.
That have marine representatives. Please correct me if I'm wrong.
This seems promising. I get that simple is better in most cases, but a simple iris shutter at the end would solve a few. Yeah. Huh? Oh, just get it in the quiver. Just get it in the quiver. Quiver's not the right word. That's a different thing. But the place, the thing.
Lauren?
Yes.
if this core is successful,
Mm-hmm.
I have a request with Armando to do a core on top of the living bivalve a little to the left. That is going to include the bivalve and the sediment down to 5-10 centimeters.
Okay, yeah, can do.
Also, no pressure. I
Or
think
number
it's one of
3.
the grad students
You got it.
running the ROV right now. So I imagine that poor grad student is sweating. I would be crying right. This would be the point where I started crying. No joke. I would be right. Ah, I think they've got it. There we go. There we go. There we go.
I think we need a round of applause for the
Yes.
the ROV pilots right about now.
Excellent. Let's go. Whoa.
It does look like a Winogradsky column.
And then YouTube farted again. Yeah,
A
fudgy, fudgy,
living
fudgy.
bivalve on top, if we can. Yeah, core number 3, please.
I'd be using words not safe for
Yeah,
Twitch
this one's
YouTuber.
I
Just
think
cut
we
his mic.
saw
Just
one
cut his
or
mic.
two.
So I think they're avoiding using.
The voices that you hear in the background, these are scientists of the and graduate students of the expedition, and we have meaningful discussions on how to pick sampling sites and what priority to have at that particular moment. So,
Oh my gosh, there's still so many fibers.
slightly to the left.
Slightly to the left of... Oh,
Oh my gosh, farted.
oh. Did that have a live bivalve on it? Where? Oh, the little guy. Oh, okay.
Oh, I like her. I was thinking
No?
they meant like a big one.
Okay. Left?
A little more left.
Left?
Right
There.
Is that one alive?
Okay.
Oh, it does look like it might be moving a little. Oh, so they're gonna try to sample. Just right over it. No stress. No stress.
Oh.
Oh, it got knocked sideways a little. Oh, that's bigger than I thought it was initially.
Hey Lauren.
Ah,
just to
it's
clarify,
in!
Did Armando mention that Drew and I want to take a core here of the film as well?
Yes.
Okay,
We're
thank you.
taking 2 more after this one. One for Daniel and one for you.
Perfect, thank you.
You are so welcome.
Yeah. Katie just did. Where?
Oh.
Just
Actually,
holding it for a
could-
minute. Let
could
it
someone
settle.
take over for me for just 5 minutes? I need a biological break. Thank you.
A biological rate.
Direct core sampling. I'll be right back.
Okay.
Here we go. Sorry, I'm taking some care of some stuff on my end. There we go. It's staying. Let's go. That is an excellent sample.
have a request for another core, number 4, on top of the white bacterial mat.
No one's listening to you, so you can just tell them. They don't have.
Let's finish with that first. Perfect. For the pilot,
What are those? More bacteria stuff. Oh gosh, there's more of it. Ah, anyway, sorry.
you have to appreciate the excitement of people trying to get us, as many samples as we can.
Yeah.
Now, again, remember, a sample, of course, it takes a huge amount of effort to be here and be able to get those samples. But the vast majority of the work is done back to the lab.
I wonder how the different scientists are rationing out samples. Does each field get X amount of samples? I think they specifically request... I think specific teams request specific samples based on what the ROV is doing for the day. I know on certain expeditions, when they launch, there's like a wish list for like the biology team and a wish list for like the geology team, and they try to cross things off as they progress. Can anyone mind if we just take a piece of the seafloor? We're not here to know, yoink. But earlier, when they were collecting the crinoids, for one person, another person was like, "Can I get 2 of those as well?" So it's very much a requested thing. And I think some interdisciplinary sharing probably does occur. Triton, Lord of the Deep Ocean, is quite displeased. We're tearing up his flooring! He just put that down. The poop floor. Yeah, this is maybe we don't want to think about this, but all of this soft sediment is marine snow, which is it's fish poop. I mean, it's other stuff as well, but it's death and fish poop. Sometimes we'll be cruising along in the ROV across what looks like beautiful white sand. It is not beautiful white sand. Oh, that's another excellent one. I guess the trick really is just to hold the sample in place for a minute. And you can see the bacterial matting. Yeah, that was a really cleanly done sample. Let's go.
Looks like a very successful core with microbial biofilm.
Oh, Stephanie Floriska, hello. Welcome. I'm sorry if it took me a while to see your message. Welcome.
Beautiful. Big thank you to the ROV pilots.
I'm betting it has quite a lot to do with how much structure there is. You can't really see in an individual sample
There's a
too.
lot of white
Very
on the top
much.
of that. I'm gonna have lots of fun.
There was one point where we were watching a dive and they just could not get a sample to stay in one area. And then they just kind of moved a bit and they were able to get a sample. So I think this is the last sample for this area?
Geology, okay.
Yeah, someone just said this one's for, so they're taking, I guess the previous ones were for biology and maybe the microbiologists. This one specifically is for geology.
Maybe here. I don't know if
You're gonna get, you see all the gray matter, that's from the bottom of the other core. So
Mhm.
it's
Oh
get
my
rid of
god,
it,
I'm
you
so
know.
sorry.
See what I'm saying? Where it's like dark gray on top of the white? You're okay with that mix. But it's okay, otherwise.
Oh, the
you
dark
can
gray,
multi-breed
it's
it.
right under my, it's right
To the
there.
left, and more portion
yeah,
of
I
it
think
too.
that is better to check out
Yeah,
another one
yeah,
here.
yeah, we can move, just see the clean patch over there. We just move right over there. No problem.
There's more, there's more.
Yeah.
But wait, there's more.
But wait, there's... This is a really good crew. Oh, I guess we're relocating a little bit to get the sample they want. Right, I think it was Javier that said there's a... As cool as the dive streams are, a lot of the work will happen in the lab later. Like processing these samples.
There, yeah, okay.
You can take
Fish. It's all fish poop. Oh, I'm sorry, I didn't see that at first. It's all fish poop, pretty much. Pretty much. You know what? Let me actually look it up so I'm not telling lies on the internet. Let's see. Let's see what the official like. What is marine snow? And Noah has an answer. As plants and animals near the surface of the ocean die and decay, they fall towards the seafloor, just like leaves and decaying material fall to the forest floor. In addition to dead animals and plants, marine snow also includes fecal matter, sand, soot, and other inorganic dust. The decaying matter is referred to
Won't be
as
able to tell you
marine
guys
snow.
on this stream, but maybe on the next stream. I do smell every sample that comes up because all 5 of our senses are really important when it comes to things. So
No! F's in chat. She's sacrificing herself
Not
for
my
science.
I am not tasting anything. I would like to specify.
Steamkey. Also ship discharges. F. Oh, yeah, big. I'm so glad she had that. But yeah, it's decomposition and poop, as well as sediment. And I imagine a lot of the decomposing stuff gets to the floor, and then gets eaten. So then it is poop. It's just all poop. I get why Scotty says it was all poop now. That's on space dust.
Technically, you can taste everything at least once, but
I worry about this crew. Why the risk? Exactly. Don't do it. Do not sample the marine snow. Don't eat the yellow snow and don't eat the marine snow. Don't do it.
not from the spot.
Decaying matter is referred to as marine snow because it looks like a little bit like fluffy, white fluffy bits. The snowflakes grow as they fall, reaching several centimeters in diameter. Some flakes fall for weeks before finally reaching the ocean floor.
We will know how it smells by tomorrow when we process the cores. We'll report back.
We'll report back! Continuous rain of marine snow provides food for many DC creatures. Many animals in the dark parts of the ocean filter marine snow from the water or scavenge it from the seabed. NOAA scientists and others have measured the amount of usable material in marine snow and found that there's plenty of carbon and nitrogen to feed many scavengers in the deep sea. A percentage of material is not consumed in shallower waters. It becomes incorporated into muddy ooze, blanketing the ocean floor, or is further decomposed through biological activity. About 3/4 of the deep ocean floor is covered in this thick, smooth ooze. Ocean exploration was a mistake!
The non-Dr. Warren.
The ooze collects as much as 6 meters. 6 meters every million years. Alright. That's 6 meters every million years. Anything can be tasted once. Fluorine that doesn't have an oaky aftertaste. You'll survive long enough to find. Will you survive long enough to find out? Please do not. Chat, please do not try anything just once.
Taking over for Lauren. I'm Drew Palmer.
Oh, someone in their chat is like, I've talked with my co-advisor about eating fresh specimens of the group I work with, and she was like, no, Wolf. Yeah, it's wild. It makes sense though that it floats very. My hands are just doing things. Floats very slowly. Disco says hydrate. Let's hydrate everybody.
Okay. All right, Lauren is back.
She's back.
Lauren is one of the astrobiologists.
Was it good to be the king?
She stepped away
It's all good.
for
I'm
biological
more than willing
reasons.
to give this seat up in a little bit so that I can go eat something. Haven't had my dinner yet.
Oh gosh, it's nearing time for me to wrap up. Although I did start a little late, I should probably end on time. I have no intention.
You
of
are
doing
popular,
more.
Dr. Drew.
So I won't be eating anything that we take from here, but in my work off of the ship, we grow different crops in simulated Martian regolith, and I can say I have actually tasted tomatoes that have grown out of regolith. So there are some times where it is okay for us to taste our work. I just don't think this is one of them.
That's
In this
fair.
case,
That's fair.
Do not lick the science.
Do not
lick
lick the science. Don't lick the.
Unless you see a geologist do it first, I guess.
Only Oscar.
Oh no, they discovered the haunted shipwreck while I was gone? This is why you never take bathroom breaks.
If the geologists start licking rocks, we're in trouble. That's why we never leave them alone.
No. You need to be supervised.
At all times.
This crew is excellent. I love them. Okay, let me lower the volume on this because something in chat needs to be addressed. Something in chat needs to be addressed here real quick. Brick, what do you mean by I have no intention to try murder hornet sting twice? Elaborate. What do you mean twice? Brick, did you get murder hornet? Brick, what? Brick, wait! How? Brick! Unlimited free shrimp. You don't want the shrimp. There's a reason it's free.
Gherkin.
Do not lick the science, but how am I going to know what dark matter tastes like?
Color. This is so silly, but it reminds me of like a green velvet sofa. Like if I don't know why it makes me think of that.
All right, we can resume the dive.
Yeah.
You said don't do anything twice. Okay, you have. Okay, all right. I was like, Brick, how did you manage to get?
Can we stop
How did you
and
manage to get
grab
stung by a
crab?
murder hornet? But okay, I got it. I got the joke now.
I'm
It was
guessing with the suction.
Brick was building on my joke, but
Probably
I'm
gonna be the
stupid,
easiest
so I
way to do
forgot.
that.
You don't want social media either, and it's free. True. It's free because you are the product. I'm gonna do it, chat. I'm gonna build the cringe anime forum. We have to return. We have to return to the early 2000s. I'm going to lick the science. Don't lick the science. I was thinking, like, what science could I lick? I could lick the quail. Like, no, don't lick the quail. Oh my gosh, what you chirping for? What you chirping for?
Yeah, it's fine.
What you
Got one.
chirping? What you chirping? Yeah? Get in the nest, silly.
Crab
Yeah,
grab.
get in there.
Operation crab grab. I know it's a lobster.
To be fair, there's a part of me that would really like to return to the early
That's news
2000s.
to me. I didn't know that.
Yeah.
I believe.
Oh, Operation Crab Grab is real. We're grabbing the crab. Getting slurped. Get slurped.
That would make sense.
Friend.
I do like crab and lobster.
Operation Crab Grab is a go.
For the squat lobster.
Yes, it's going to. It swims butt first, so.
All right.
Booty first.
Oh, there.
Oh
no! Operation Crab Grab has failed. We'll get him next time. It's next time, round 2. Let's go.
Manipulators and Tyler is flying at the moment. The chat wanted to know.
He's on to us.
Look at that shrimp. He looks like a Muppet.
We're crabbing the grab. Yeah. Wait. The early 2000s are lousy. Early 2000s internet was one of the few good things. There were things about it that were really good. Also appreciate all. Yeah, the internet I think is better overall. But I'm definitely not the first person to decry the fact that there's like 5 websites now, and nobody uses forums, and all the information is locked into discords. It's not great.
Yeah, Moriel needs me.
Murder hornet nest in my mansion. Twindle and Nermi.
Alright. This time I'm back for real.
I did not know about murder hornets until later, but the landlord was thankful for. Oh no! Yeah, forums are. They're not completely dead yet. But they're definitely more rare.
Crafty crab.
Window, not. Got it. I was.
We just want to be your friend.
No! Do you live in a Takahashi series? Brick lives in the Curry Mines in Maine, which explains things. You know what? Brick would be a good Rumiko Takahashi character. Tone indicator positive, but also kind of chaotic.
2 hours ago, 2.5 hours ago now at this point, we actually identified this fairly sizable area that appears to be
Crab grab? Crab is grabbed! Crab is grabbed! Oh.
Right. And is that going into sampler 1?
Oh!
Oh.
He's making a break for it! He's playing the suck!
I'm watching the jar cam.
Has science gone too far? Pretty much, yeah. Crab is grabbed! Crab is grabbed! Oh, we're waiting for crab to drop into the sample box up there. Any minute now.
It just disappeared.
Oh no. No!
It's stuck in the
No,
sampler.
it's stuck in the...
He's holding himself in the tube around the metal ring.
No!
Yeah. So please.
I think they're trying to suck up some sediment to get him to let go. Oh!
No, he's still there.
He is a... Oh my gosh, YouTube, I'm gonna fight you. Alright. Ignore that.
Oh,
We're
perfect.
not
Thank
seeing.
you.
No, he's still there. I can see him. I can see his face.
No! Perhaps we were too ambitious with the crab grab.
Yeah.
Huh?
Alright, so while we were working on getting this crab, I'll just again say we about 2.5 hours ago identified what appears to be a declining methane seep, but it is an active one. And so that was one of the main goals of this expedition, and so here we are. We've been on a dive since about 6 o'clock this morning, trying to identify one of these, and we
Oh
found
no,
a
is it
mound
really
here that
stuck?
appears to have a fair number of them.
Oh no.
And currently we're doing a little bit of sampling, but we're starting to explore the area a little more in depth while we capture a few sediment cores here to look at the microbes and whatever metabolites might be leaving behind, but also trying to catch some of the larger fauna of the area.
I believe they
We got a lot of excitement in
can
here.
reverse the. I'm actually not sure. That might be his home now. Which would kind of suck because they've got several more hours in this
I believe there probably was one, and I don't think I know. So if somebody in here wants to go ask. Sounds good.
just shake it. Just shake him loose. Oh man, no, bro is in there.
Craftiest crab that ever lived.
Yeah, he's got himself braced against the.
Is the tube able to eject? Could you eject into a bio box?
So that's what Kaven was asking: is can they eject? Get the unsuck machine. Can we reverse suck? I think they are going to. Okay, I think they can eject. I think they're going to eject into a bio box. Like she suggested. That's a blow. It's not unsuck. It's a blow. Got it.
Eject it. I wonder. Would it be possible to try ejecting it into a bio box?
That's a good question. I think the problem is it's holding on pretty tight.
Bruh!
They were wondering if you can eject it into a bio box. Can you eject it into a bio box and reverse suction it? There is no reverse suction. There's no reverse suction.
Okay.
There is no reverse suction.
Actually, the
There
tube
is
not
there
capable
is
of
no
reverse
blow.
suction.
Well. All of the other samples will have to
You
be
two
around
options.
this crab.
Suction, no suction. That's what Hannah in the room just said.
Can you hover over the bio box and then turn off suction and then like shake it?
He's
Well,
holding on.
they've been turning the suction off, and it's just not going
Yeah,
anywhere.
He's just holding on.
Hey!
Oh! I'm not
I
doing
don't see
that.
it in
That's
the
on
jar
their
yet.
end. You can
There's
still
a lot
see
of
the.
sediment in there.
I'm a happy crab in a cave. This is what I evolved for. That's a serious design flaw.
He's trying
I
so
mean,
hard.
human got abducted by alien. Yeah. Now he is stuck only even deeper. Oh no! Oh, no, you're gonna have to take the hose apart to get him out. Quails be eepin'. Quails be peeping. Crab really downloaded the mothership Zeta.
That's okay.
Looks like he's in a bit of a pinch.
Hey.
We're getting a pilot change. So, with
Gosh darn it. Did it die again?
Is Caitlin Hubrick. I actually study underneath Dr. Palmer, the one who just introduced me. I am an astrobiologist. I
So
am
someone
currently
just
working
got fired.
on
No,
my
no.
master's degree.
They do ships.
My
They do.
research is primarily focused around sustainable space-based agriculture, but I also have interests in desert microbiology, as well as life in other planets and moons in the solar system, which is why I'm here on this trip. I'm gonna pass the microphone off to our next grad student.
Hello, my name is Amber Wu. I'm a PhD student studying prebiotic chemistry on the origins of life. My work centers on experimental and computational chemistry, looking at how coasphere droplets can be a precursor or, I guess, model for cell membrane, cell compartmentalization before lipid membrane forms.
and Miguel.
If Miguel, introduce yourself to the viewers at home.
Let's pass her back.
All right, and next up is Miguel, who's also my data logger today. There's far too much joviality in this room.
Y'all are too happy. Tone it down. But no, no one got fired. They do regular shift changes to ensure that everyone gets proper amounts of rest because they are on that ship 24/7. So staying rested is very important. Gonna head out, see. Yeah, Bear of Peace. Thank you for coming in. Aside from the heating pad, have there been no quail scares? No, everyone seems to be okay. I have been feeding them additional, like, extra nutritious foods. Oh, that was my hands, man. And, early on, I was, more or less hand feeding, quail, but they are all strong and alert now. So it's just a matter of making sure that the smaller ones don't get, like, pushed out of the way for food. I haven't been doing it while I'm streaming today, but Beta will wake up like twice for every time that Delta and Lambda wake up once. So sometimes I will wake up Lambda and... Wait, no. Delta will wake up twice for every time B and Lambda wake up once. So sometimes I'll wake up B and Lambda. To make sure they're eating. Like, I'm pretty sure that's Delta just literally bouncing off the walls right now. And B and Lambda are just like, nah, it's time for naps.
Okay.
I want to see at least one severe sleep-deprived,
Miguel,
overcaffeinated, full-blown cat.
Basically, right now, just make a note that the crab is in the suction sampler. We can still use it
Wait,
to
did it just
bring
fall
other
through?
stuff across, but he's somewhere in the tube, so we'll just be on the lookout.
I'm going to draw a tube, ROV setup.
Moving forward up this ridge, we think there may be
Alright.
even more methane seeps
On
here.
that note,
We've
we
still
are
got
gonna
several hours left planned on this dive, so we're going to continue pushing forward and see if we can find some other sites, and then make our way back here to collect more. Okay. There you
I'm
go.
also not sure if I've seen Lambda drink
Not
water.
like all the way up a little.
Okay, so they are going to continue. I'm going to drop a link for them, for you guys, in case you want to watch the dive continue. I unfortunately do need to stop. It is time It's time to stop. Where are my parents? So let me, like I said, let me get you this link. Let me see if I can raid them on Twitch, actually. On Twitch we will raid, unfortunately. Unfortunately on YouTube, it will just sort of end. But there's the link for where they will continue the dive. Oh my gosh, just chirping. Little chirpy babies. Chirpy little children. I will look into Plant the Moon more. It does, I may have pulled up the website, on another monitor, to just kind of take a look at what all is going there. It does seem to be primarily a high school thing. But I'm curious if I would be able to do it. Purchase a Plant the Moon registration pack or apply for grant funding to cover the cost of one of our partners. Oh no! So it might... How much is this? How much is Plant the Moon? Oh, it's $200! We might not do this. Never mind. Each team is required to have one adult coach, typically a teacher. It can also be a parent or older. Yeah, this does seem like it is aimed primarily at high school students. So as cool as that is, that might not be something we'll do. Donation goal. I still have, I have... Chat, you should buy me $100 of shale from Utah off of Throne. So that I can search for trilobites live on stream. When they mail me 50 pounds of shale from Utah. Is it 50 pounds? I don't remember anymore. Yeah, alright. I understand why they do that, but all the same, I often feel like that should be an adult category for grown. Yeah, like... You know what? If they're... I'll look into it more. But I think doing it as an adult, or just someone passionate about this kind of thing would also be really interesting. One coach may register as many teams as they like, 1 to 5 students per team. Yeah, this does seem very... This does seem very, Yeah, you have to be between the ages of 13 to 19. Ah, rip! Ripperoni. Ah, well. Highest school of dumb stinky borbs. Don't science good, yes. I am curious about more things like this, though. Yeah. Anyway. How old is that in quail years? Oh man, quail only live for like 5 years, so... I don't know, like 3 months? Like... Anyway, rate! Sorry, we're rating. I'm sorry, I am stinky. Poochie, hello, welcome! We are unfortunately just about to end. Let me see if I can raid Schmidt Ocean. I think they probably still have raids turned off, but let me check anyway. Let us raid you. 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 No! They still don't accept raids! Schmidt Ocean, turn on raids on Twitch. Do it. Do it. Alright, so we'll raid another VTuber, probably. Actually, wait, I have to see if we were talking about fossils. I have to see, if the, paleontologist I like to raid is live. No, he's not. Alright, we'll raid a VTuber. We're gonna raid Will, who's doing art. My VTuber rival, who's scared of the ocean. We could all run in there and make the request. No, I don't want to do anything that could be construed as like causing problems. The person who runs their like streams is also like trying to do clips, and they already have a full plate. I would hate to cause any kind of trouble. Okay, so that's said. And I will say thank you so much, everyone, for coming to this stream, watching this dive. This was a really cool dive. And I will see you all later. Yeah, goodbye for now, everybody. Thank you so much. Wah, wah, wah.